Welcome to the deep dive. In a world absolutely swimming in information, it can feel incredibly challenging to move beyond just skimming the surface, right? To truly master a complex topic.
Yeah, we've all been there. You read something, think you get it, but then try explaining it.
Exactly. And that's why we're here. This isn't just, a quick summary. It's really your pathway to getting a professional level grasp of these intricate subjects. We want to unearth those surprising connections, those profound insights that, well, genuinely elevate your expertise.
And that's our exact mission for today. We're embarking on a really focused journey into the intricate world of viruses.
Right.
Specifically, we'll be undertaking an exceptionally thorough deep dive into the human retroviruses. We're drawing heavily from Chapter 201 of our source material for this.
Okay.
But to really provide a complete picture, a good foundation, we're first going to examine some critical antiviral treatments, specifically for influenza. They elegantly illustrate these core principles of how we actually combat these microscopic invaders.
So the goal isn't just to tell you stuff, it's to equip you with the kind of knowledge that lets you think like an elite professional in this field. We're going to break down these dense scientific concepts, digging into every crucial detail, uncovering those critical nuggets of knowledge, those surprising insights.
Really getting into the weeds of the source material.
Exactly. So you walk away with a comprehensive, really nuanced understanding.
And why does this level of detail matter? Why should you care? Well, truly mastering these fundamental biological mechanisms, it isn't just for scientists in a lab coat.
Right.
It illuminates the very behavior of diseases. It reveals how our own genetic information can be, well, profoundly influenced, even manipulated. And it opens the door to groundbreaking fields like gene therapy.
So prepare for some serious aha moments, I think.
Absolutely. Because what we're about to unpack will fundamentally reshape how you perceive biological information flow and, our ongoing, very sophisticated battle with pathogens.
All right, sounds fascinating. Let's start by setting the stage then. Diving right into the front lines of defense against viral infections, beginning, as you said, with antivirals specifically for influenza.
Good place to start.
So when we talk about fighting the flu, one of the key drug classes are these neuraminidase inhibitors. Sounds technical, but the way they work is actually pretty clever. So what exactly is neuraminidase and how do these drugs mess with it?
Okay, so neuraminidase, it's a vital viral enzyme. Think of it like a molecular key or maybe tiny scissors on the surface of the influenza virus particle.
Okay.
Imagine new influenza virus particles, freshly made inside an infected human cell. They're essentially stuck there initially.
Trapped.
Exactly. They need to escape to go infect other cells and keep the infection going. This neuraminidase enzyme, it acts like those tiny molecular scissors, cutting the bonds that tether these newly formed virus particles to the surface of the infected cell.
Ah, so it snips them free.
Precisely. This allows them to effectively bud off and spread freely throughout the body, infecting more and more cells.
Okay, so if you can block or inhibit that enzyme, you're basically tying the new viruses to the cell they just came from, preventing their escape and slowing the whole infection down.
That's a perfect way to put it. By inhibiting neuraminidase, these drugs effectively limit the egress, the exit, the release of new influenza virus particles from an infected cell.
Right.
This action dramatically slows the spread of the infection within the body. And, this isn't just about feeling a bit better sooner. It's crucial for reducing the overall viral load, diminishing symptom severity, and potentially preventing more serious complications down the line.
That makes incredible sense. It's like throwing a wrench in their escape plan. Now, when it comes to actually using these drugs, is there a critical time window? I always hear that timing is absolutely key with antivirals.
Timing is absolutely critical, yes. There's a very clear golden window. These drugs are definitely most effective when influenza illness is recognized and treatment is initiated very early on.
How early?
Well, ideally, this means either getting a confirmed diagnosis through, say, rapid diagnostic testing, or having really strong clinical and epidemiological evidence pointing towards influenza. The sooner you start, the bigger the benefit.
Okay.
Experimental trials have consistently shown that if these drugs are given within 48 hours of infection onset, that's just 2 days from when symptoms first appear, they can speed up the resolution of symptoms by about 1 to 2 days.
Which can feel like a lifetime when you have the flu.
Absolutely. For someone really hit hard by the flu, those 1 or 2 days can mean significantly less discomfort, a quicker recovery, and importantly, a lower chance of developing secondary complications, like bacterial pneumonia.
So optimizing the outcome, not just symptom relief, but potentially stopping it from getting worse.
Yeah.
And speaking of use, do these neuraminidase inhibitors just treat active infections, or can they be used for prevention too?
Oh, they absolutely have a dual purpose, both prophylactic and therapeutic. Therapeutically, yes, they're used to treat active symptomatic infections, especially when given early, like we just discussed. But they also play a crucial role prophylactically, meaning for preventing illness altogether.
How does that work in practice?
Well, it can involve giving them throughout the flu season for individuals who are at particularly high risk of severe complications. Or, maybe more commonly, on a short-term basis when someone has had close contact with a confirmed case of influenza, maybe within a household or, a nursing home setting.
So if someone in your family gets the flu and you're vulnerable.
Exactly. If your child or spouse comes home with confirmed flu and you're in one of those high-risk groups, these drugs can be used to try and prevent you from getting sick in the first place.
That's incredibly powerful, that preventative aspect. What about those more complicated situations, though, like patients hospitalized with really severe influenza? Is there a point where it's just too late for these drugs to help?
That's a nuanced but really critical point in clinical practice. Even if that ideal golden window of 48 hours has closed, their use is still recommended for complicated influenza infections in hospitalized patients. This holds true even in situations where the diagnosis might have been significantly delayed, or even if there isn't formal, rigorous proof of efficacy in those specific delayed scenarios.
So even without perfect data for late use, the recommendation stands.
Right. The clinical judgment is that the potential benefit, even if it's reduced compared to early treatment, likely outweighs the risks in these very severe cases, especially for those patients at high risk of serious morbidity and mortality. It really underscores their role as a vital tool, even when the timing isn't optimal.
It's about giving patients every possible edge, even if the situation isn't perfect. Okay, this leads to an important question. What about the risks? Specifically, the potential for the virus to develop resistance to these drugs. Is that a major concern?
It is indeed a concern, yes. And it's a risk that's inherent to all the available neuraminidase inhibitors, the potential for resistance development.
How does that happen?
Well, the risk is particularly elevated with prolonged administration. For example, in an immunodeficient individual who might have persistent influenza virus recovery, they're shedding virus for a long time, and they're on the drug for a long time.
Right.
Think of it like this. The longer a virus population is exposed to a drug, the more opportunities there are for just random mutations to occur. If one of those random mutations happens to confer resistance to the drug,
then that version gets selected for.
Exactly. That resistant variant can then be selected for and multiply, potentially becoming the dominant strain in that individual and maybe even spreading.
That makes perfect sense, evolution in action, basically. But what's the current situation? Is resistance widespread among the flu strains circulating now, or is it more of a background worry?
Fortunately, at the moment, resistance is not widespread among circulating influenza A or B strains, which is obviously good news for their continued effectiveness.
Okay, that's reassuring.
However, it's definitely not just a theoretical lab concern. Resistance development has been demonstrated in lab settings, and its emergence in the clinical setting, if it became widespread, would indeed significantly impact the future utility of these drugs. So diligent surveillance for resistance is an ongoing and absolutely essential part of global influenza management. We have to keep watching.
Always vigilant. Okay, so for now, they remain largely effective. Good. Now, when we talk about who benefits most from these neuraminidase inhibitors, are there specific groups or demographics that really stand to gain the most protection?
Absolutely. And the defined risk groups who stand to benefit most from these drugs mirror, quite intentionally, the same groups for whom influenza vaccination is already a high priority.
Okay, so who's on that list?
It's quite comprehensive. It includes children under 2 years of age, adults over 65 years of age, patients with chronic underlying medical conditions, think chronic lung disease, heart disease, diabetes, kidney disease, liver disease, immunosuppressed individuals whose immune systems are compromised for various reasons, pregnant women, and also women who have delivered infants within the past 2 weeks.
Okay.
Also included are patients under 19 years old who are receiving long-term aspirin treatment because of the risk of Ray's syndrome if they get influenza. Other important groups are Native Americans, including Alaska Natives, who historically have higher rates of severe outcomes, morbidly obese individuals, and residents of nursing homes or other chronic care facilities where outbreaks can be devastating.
That's a very clear list, really highlighting the most vulnerable populations.
Okay.
And you mentioned something interesting earlier that even if someone in these high-risk groups has been vaccinated, we might still consider using these antiviral drugs. Could you elaborate on that sort of layered approach?
Yes, this is a crucial point. It really underscores the multifaceted strategy we need for influenza management. Vaccination is, without doubt, the primary preventative measure. The goal is to prevent infection entirely, or at least significantly reduce its severity.
Right, the first line of defense.
Exactly. However, for selected high-risk cases, even if they have a history of recent vaccination, the use of neuraminidase inhibitors should still be considered if they get infected.
Why is that?
This layering of protection is vital because, well, vaccine effectiveness can vary from season to season and even within a season for different individuals. High-risk individuals might still experience severe illness even if they've been vaccinated, especially if their immune response wasn't optimal, or if a vaccine mismatched strain happens to be circulating predominantly that year. The antivirals provide an additional targeted layer of defense in those situations.
A layered defense. I like that framing. Okay, let's get into the specifics of the available neuraminidase inhibitors themselves. You mentioned oral oseltamivir, nasal spray zanamivir, and intravenous peramivir. Can you walk us through their unique characteristics? Let's start with oseltamivir, which you said is the most widely used.
Certainly. Oral oseltamivir, perhaps better known by its brand name Tamiflu, is indeed the most widely used. It's an orally absorbed drug you take it by mouth, typically as a pill or liquid suspension.
Okay.
But it's important to understand it's what we call a prodrug,
Meaning?
meaning the drug itself isn't actually active when you swallow it. It needs to be converted in the body to become active. Specifically, it's efficiently converted to its active component, called oseltamivir carboxylate, primarily in the liver. This activation step is absolutely essential for it to work.
I see. And what about side effects?
The most common side effects are primarily gastrointestinal. Nausea is probably the most frequently reported one, sometimes vomiting.
And are there specific recommendations for its use, like certain age groups or situations where it's preferred?
Yes, there are some specific recommendations that make it a go-to choice in certain scenarios.
Yeah.
Oseltamivir is generally considered the preferred agent for the treatment of pregnant women, which is a significant point given the potential severity of flu during pregnancy and its established safety profile in that group.
That's important.
It's also approved for treatment at basically any age, starting as early as 14 days of life in infants. This makes it exceptionally versatile across almost the entire age spectrum.
Okay. Any contraindications?
Well, the obvious practical one is that poor oral intake or problems with absorption would be a contraindication for taking it by mouth. However, even in those cases, like someone who can't swallow well, it can still potentially be given via an oronasal tube to ensure it gets absorbed systemically.
And for hospitalized patients?
For hospitalized patients with suspected or confirmed influenza, oral or enterically administered oseltamivir is recommended to be started as soon as possible, really reinforcing that urgency we discussed earlier for achieving the best possible impact.
Understood. Okay, so what about the nasal spray version, zanamivir? How does its delivery method change things, and who usually gets prescribed this one?
Right, so nasal spray zanamivir, marketed as Relenza, uses a fundamentally different delivery method. Why? Because it's not orally bioavailable, meaning it doesn't get absorbed effectively into the bloodstream if you just swallow it.
Ah, okay.
Instead, it's administered as an inhaled dry powder. It's dispersed through a specialized device called a discaler, which delivers the drug directly into the respiratory tract, the lungs, and airways.
So direct delivery to the site of infection.
Exactly. But because it's inhaled, there are important contraindications to be aware of. Patients with pre-existing respiratory conditions like asthma or chronic obstructive pulmonary disease, COPD, are considered relative contraindications.
Why is that?
Because there's a potential risk of bronchospasm, basically, airway narrowing, or irritation triggered by inhaling the dry powder. So it's generally avoided in those patients. Age-wise, it's approved for treatment in persons 7 years and older.
That delivery method difference is really key for picking the right drug for the right patient. And finally, what about intravenous peramivir? When does that come into play?
Intravenous peramivir, sold as Repivab, offers a distinct advantage because it's given as a single intravenous infusion directly into the bloodstream.
So bypasses the gut entirely.
Correct. It's typically considered for patients who cannot tolerate or effectively absorb oral or enterically administered oseltamivir. So think about a patient who is critically ill, maybe unconscious, or experiencing severe vomiting, or has significant gastrointestinal issues that would prevent oral absorption. In those cases, peramivir offers a reliable alternative route, ensuring the drug gets into the systemic circulation effectively. It's licensed for individuals aged 2 years and older.
And for all these neuraminidase inhibitors, oseltamivir, zanamivir, is there a standard treatment duration?
For oral oseltamivir or intranasal zanamivir, the usual duration of therapy is pretty straightforward. It's typically 5 days, with dosing usually occurring twice a day. This standardized regimen helps ensure effective viral suppression and symptom resolution.
Okay, that covers the established neuraminidase inhibitors really well. But medicine is always moving forward. What about newer antivirals? I've heard about baloxavir marboxil or XOFL-UZA. How does that fit into the picture?
Ah, yes, baloxavir marboxil, or XOFL-UZA. It represents a truly significant advance, really a novel approach to treating influenza. It got FDA approval back in 2018 as a first-in-class compound for people 12 years and older.
First in class, what makes its mechanism different?
Well, unlike the neuraminidase inhibitors, which, as we discussed, target the virus's exit strategy,
Right, cutting the ties.
baloxavir targets a different, much earlier stage of the viral replication cycle. What's fascinating here is that it inhibits an enzyme called cap-dependent endonuclease.
Okay, what does that do?
So imagine the influenza virus trying to set up its replication factory inside your cells. It needs to hijack your cell's machinery to make its own messenger RNA the instructions for building new viral proteins. This cap-dependent endonuclease is a crucial viral enzyme that enables the virus to basically steal necessary caps from your own cellular messenger RNAs. It's a process called cap snatching.
Cap snatching. Sounds sneaky.
It is, and it's essential for the virus to initiate its own gene expression. By inhibiting this specific enzyme, baloxavir effectively shuts down viral gene expression right at the start, preventing the virus from even making its building blocks. And it works against both influenza A and B strains.
And I heard it's just a single dose. That sounds incredibly convenient.
That is absolutely one of its most significant advantages and a major draw for patients. It's a single-dose oral formulation. That dramatically simplifies adherence compared to taking pills twice a day for 5 days.
Big difference.
Huge difference. And in clinical studies, when it was given within that same 48-hour window of symptoms starting, baloxavir showed impressive efficacy. It decreased symptom duration, reduced viral shedding, meaning how much virus the person releases, and even lowered the rates of subsequent antibiotic use in healthy individuals with uncomplicated flu.
Lower antibiotic use suggests fewer secondary bacterial infections.
Exactly. That points to its ability not only to shorten the illness itself, but also potentially to reduce the risk of those dangerous secondary bacterial complications that can follow influenza.
So it sounds like a real game changer for certain patients. Are there any downsides or limitations, similar to the resistance concerns with the other drugs?
Yes. As with pretty much all antivirals, the potential development of resistance is a concern, and it's something that's continuously monitored for baloxavir as well. But crucially, there are also limitations based on current data. Because there's currently a lack of sufficient information for certain specific groups, baloxavir is not recommended right now for pregnant women, breastfeeding mothers, outpatients who have complicated or progressive illness, severely immunosuppressed patients, or hospitalized patients.
So it's mainly for uncomplicated flu in otherwise healthy people right now.
Essentially, yes. It highlights that while it's very promising for those straightforward cases in generally healthy individuals, its use is still more restricted to those specific scenarios until more extensive data become available for these more vulnerable populations.
So not quite a universal solution yet, but definitely a significant targeted addition to the antiviral toolkit. Okay, moving beyond these specific antiviral drugs, what other aspects of comprehensive influenza treatment are absolutely essential for good patient care?
Oh, absolutely critical. Beyond the antivirals, essential supportive measures are just paramount for managing patients effectively.
Like what?
Things like diligent maintenance of fluid and electrolyte balance. People with flu can get dehydrated easily from fever and maybe vomiting. Oxygen supplementation is key for patients experiencing respiratory distress or low oxygen levels, hypoxemia. And, effective fever control with non-steroidal anti-inflammatory drugs, NSAIDs like ibuprofen or acetaminophen, can improve comfort and reduce the body's metabolic demands.
And what about the risk of those secondary infections, especially bacterial ones? You mentioned flu can pave the way for them.
That's an extremely important point. Can't stress it enough. It's crucial to be vigilant in monitoring for and then promptly treating any suspected secondary bacterial complications with appropriate antibiotics.
Why are flu patients so vulnerable?
Well, influenza can significantly compromise the integrity of the respiratory tract lining and also suppress parts of the immune response. This makes patients particularly vulnerable to bacterial invaders like Streptococcus pneumonia or Staphylococcus aureus. And these secondary bacterial pneumonias can be really dangerous, even life-threatening.
So watch out for pneumonia.
Definitely.
Yeah.
And finally, one last thing. Appropriate respiratory isolation of patients, especially in hospitals or clinics, should always be rigorously practiced according to local guidelines. This is vital to prevent the further spread of the influenza virus to other patients and, of course, to healthcare workers.
Got it. Okay, this detailed discussion on influenza antivirals provides an excellent, really practical foundation. It shows how we can precisely target specific viral mechanisms to disrupt their life cycle. It's a great example of, advanced precision medicine in action.
It really is.
But now let's pivot and really deep dive even further. Let's explore a truly fascinating, historically revolutionary class of viruses, ones that fundamentally challenge our understanding of biology itself, the human retroviruses.
Indeed. And prepare for quite a profound paradigm shift in how we traditionally thought about the flow of genetic information in living systems.
All right, retroviruses, this family, the Retroviridae, they sound extraordinary. What's the basic definition, and what is it about their replication cycle that makes them so unique and, as you said, completely paradigm shifting?
Okay, so retroviruses are a large family of viruses, and they primarily infect vertebrates, including us humans. Their unique replication cycle is really the core of their extraordinary nature, and it's the aha moment you mentioned earlier.
Right.
You see, until their discovery, the long-held central dogma of molecular biology stated very clearly that genetic information flowed in just one direction, from DNA to RNA to protein.
Just standard flow.
The standard flow. However, retroviruses literally turn this completely on its head. Their genetic information inside the virus particle is encoded by RNA, not DNA. And here's where it gets really interesting. They carry a special enzyme called RNA-dependent DNA polymerase, more famously known as
Reverse transcriptase, that's the one. The enzyme that totally revolutionized our understanding, right?
Precisely. This reverse transcriptase enzyme directs the synthesis of a DNA form of the viral genome using the viral RNA as a template. But it does this after the virus has infected a host cell.
So backwards from the usual direction.
Exactly. So the retrovirus name isn't just, a catchy label. It denotes this revolutionary concept. Information in the form of RNA is transcribed back into DNA inside the host cell. This sequence dramatically overturned a central dogma of molecular biology, that long-held, seemingly unbreakable belief that genetic information flowed strictly unidirectionally. It was a monumental discovery back in the 1970s that completely reshaped our understanding of genetics and virology.
Wow, that's huge. Not just a niche finding, but something that fundamentally altered our view of the basic rules of life. What were the broader biological insights that came out of this groundbreaking discovery?
Oh, the implications were profound and extended far beyond just the direction of genetic information. This discovery was absolutely critical for revealing the viral etiology of certain cancers. It demonstrated, really for the first time, that viruses could directly cause malignant transformation by inserting their genetic material into a host's genome.
A direct link between viruses and cancer.
Yes. And it also introduced the concept of oncogenes. These are normal host genes that, when they get altered or maybe activated by viral integration or activity, can lead to uncontrolled cell growth and cancer. So in essence, it revolutionized our understanding of molecular carcinogenesis, showing a direct mechanism linking viral infection to the development of cancer.
So these viruses aren't just simple invaders, they're incredibly sophisticated genetic manipulators. And you mentioned they can even facilitate rapid evolution.
Yes, indeed. Beyond their impact on cancer biology, retroviruses also revealed just how easily genetic material could be transferred and integrated. Their ability to insert into the germline genome of the host cell means they can actually behave as transposable or movable genetic elements over evolutionary time.
Like jumping genes.
Sort of, yes. This unique capability allows them to potentially activate or inactivate host genes located near their integration site, which could have profound effects on cellular behavior and even long-term evolution. Furthermore, they possess this remarkable capacity to rapidly alter their own genome through processes like recombination and high mutation rates, especially in response to selective environmental pressures like the host immune system or antiviral drugs.
That adaptability must make them very difficult targets.
It absolutely does. This inherent adaptability makes them formidable pathogens and helps explain why developing truly long-lasting treatments or effective vaccines against some of them, like HIV, can be so incredibly challenging.
And what makes their disease-inducing mechanisms unique compared to, say, most other viruses we encounter, like the flu virus we just talked about?
This is a really crucial distinction that sets retroviruses apart. Most viral diseases primarily result from tissue destruction, right? Either the virus directly kills host cells or the host's own immune response to the infection causes collateral damage.
Okay.
And while these direct and indirect destructive mechanisms certainly play a role in retroviral infections too, retroviruses possess additional distinct mechanisms for inducing disease that are often more insidious and long-term,
Such as
the direct malignant transformation of an infected cell into a cancerous one, which is fundamentally different from just simple cell lysis or killing. And perhaps most profoundly, they can induce a chronic immunodeficiency state. They do this through the selective destruction or dysfunction of specific immune-competent cells. This, in turn, renders the host highly susceptible to a wide range of opportunistic diseases, both infections and certain cancers. HIV leading to AIDS is, of course, the quintessential and devastating example of this mechanism.
That's a fascinating and frankly scary duality. Direct cancer causation on one hand and systemic immune destruction on the other. It truly highlights their unique and complex impact. Okay, let's shift gears slightly and look at their physical structure and their intricate life cycle, because understanding this elaborate dance of replication is absolutely key to understanding their power. What does a retrovirus particle, a virion, actually look like up close? What are its basic architectural components?
Well, all retroviruses share a remarkably similar architecture in their virion structure, their genome organization, and their mode of replication. They're quite small, generally spherical particles, typically ranging from about 70 to 130 nanometers in diameter.
Tiny.
Very tiny. On the very outside, they feature a lipid-containing envelope. And interestingly, this envelope is actually derived directly from the host cell's outer membrane during the budding process when the virus leaves the cell.
So it wraps itself in a piece of the host cell. Camouflage.
Exactly. It's a clever strategy.
Yeah.
Inside this envelope, there's a precisely structured icosahedral capsid that's a protein shell, kind of like a geodesic dome. And within that capsid, there's a dense inner core, which is really the heart of the viral machinery.
And what's packed inside that core? I assume that's where the viral blueprint and all the key machinery for the takeover reside.
Absolutely. The core is densely packed with all the essential components needed for the initial stages of replication. It houses two identical copies of the single-strand RNA genome, which makes them diploid having two copies.
Why two copies?
That's a great question. It's thought to provide genetic stability, allow for recombination between the two strands, which increases diversity, and perhaps act as a backup. These RNA molecules are quite long, typically 8 to 10 kilobases. Crucially, these RNA molecules are complexed with that essential reverse transcriptase enzyme we talked about.
Ready to go.
Ready to go. Along with specific host tRNA molecules that act as critical primers to kickstart the reverse transcription process. And other viral proteins, like integrase, which is the enzyme that helps insert the viral DNA into the host genome, are also components of the virion particle, all prepackaged and ready to spring into action immediately upon infecting a new host cell.
So they come fully loaded with all the specialized tools they need for that initial hostile takeover. And you mentioned their RNA has features similar to our own messenger RNA, but with a critical exception, right?
Yes, this is a very interesting point where the retroviral RNA shows its kind of dual nature. It does possess features typically found in our messenger RNA, or mRNA, which normally serves as direct templates for protein synthesis in our cells. Like it has a cap structure at the 5' end of the molecule, which is important for initiating mRNA translation by host ribosomes. It also has a polyadenylation, polyA tail site at the 3' end, which generally influences mRNA stability and turnover. So structurally, it looks very much like it's ready to be translated directly into protein.
But it's not.
However, here's the crucial exception and the whole retro part again. Despite these mRNA-like features, the retroviral RNA is not directly translated into protein upon entry into the host cell. Instead, its primary fate is to be reverse transcribed into DNA. This double-stranded DNA form of the retroviral genome is given that special name, a provirus. And it's this provirus that then permanently integrates itself into the host cell's own genetic material.
Fascinating. So the RNA itself isn't the immediate blueprint for proteins. It's a template for a DNA intermediate that then becomes essentially part of us. Okay, that leads perfectly into their replication cycle. Our source material breaks it down into two critical phases, shown in figure 201-1. Let's break down phase one, the viral takeover phase, which you said is entirely dependent on the virus's own prepackaged machinery.
Right. In this first phase, it's all about the virus making its initial move and establishing its foothold inside the host cell. It begins with the virus binding to one or more specific cell surface receptors on the host cell.
Like a key fitting into a lock.
Exactly. This binding event is highly specific and effectively determines the virus's tropism, which specific cell types it can successfully infect. Following this binding, the viral envelope fuses with the host cell membrane, and the viral core containing the RNA genome and those essential enzymes enters the cytoplasm.
It's in. Now what?
Once inside the cytoplasm, the crucial work begins. The viral RNA and the reverse transcriptase enzyme, which came pre-packed in the virion, work together in a complex series of steps. This is where the magic, or maybe the menace, of reverse transcription happens. They synthesize a double-strand DNA version of the single-strand RNA template. This newly formed DNA is the provirus, now ready for its next critical step.
And then this new provirus doesn't just float around in the cytoplasm, it actually moves into the host cell's command center, the nucleus, and permanently integrates itself into the host cell's own DNA. Is that right?
It's exactly right. The newly formed provirus, guided by another prepackaged viral enzyme called integrase, then migrates into the host cell nucleus, and there it permanently integrates itself into the host cell genome. This integration is a defining and essentially irreversible hallmark of retroviral infection, making it distinct from many other types of viral infections where the genetic material might remain separate.
And where does it integrate? Is it a specific spot?
Well, while some animal retroviruses might integrate into very specific sites, human retroviruses like HIV and HTLV generally integrate pretty randomly throughout the host genome. This randomness itself can have consequences, as we'll discuss later. But the key point for this phase is that the entire process, from initial binding and entry through reverse transcription and culminating in permanent integration, relies entirely on gene products that were already contained within the virus itself as it entered the cell. It doesn't need the host cell's active machinery for these initial crucial steps of establishing residency.
So once it's integrated, it's basically hijacked the host's fundamental genetic machinery. It's become an indistinguishable part of the host's DNA. That brings us to phase two, where the host cell essentially becomes a viral factory, although under the retrovirus's strategic influence. How does the second phase unfold?
Right. Once the provirus is successfully integrated into the host genome, it essentially becomes a permanent resident gene. Now, it acts just like any other host gene in many ways. It gets transcribed into viral RNA molecules using the host cell's own machinery, specifically its RNA polymerase II enzyme.
So the host does the work now.
The host does the transcription work, yes. Though often this transcription process is heavily influenced and controlled by the retrovirus's own regulatory gene products, which finely tune the process, sometimes dramatically ramping it up. These newly transcribed viral RNA molecules then have different fates, as shown nicely in figure 201-1b of our source.
What are those different fates?
Well, some of these full-length viral RNAs are exported directly out to the cytoplasm, where they serve as the genomic material for the next generation of viruses that get packaged into new virions. Others are selectively spliced by the host cell's splicing machinery. This creates shorter messenger RNAs that encode specific viral proteins, particularly the envelope polyprotein needed for the virus's outer code. And still other full-length RNAs are translated directly in the cytoplasm to produce the essential GAG and POL polyproteins.
That sounds like a highly efficient way to use their limited genetic material and control protein production levels. And the source mentioned an interesting trick for making the POL protein, something about a stopped codon.
Yes, it's quite a clever mechanism of genetic economy and regulation. You see, there's typically a stop codon in the messenger RNA sequence located right between the GAG gene and the POL gene. Normally, a stop codon halts translation, tells the ribosome to stop making protein. However, in a precise fraction of these viral messages, this stop codon is deliberately bypassed. This happens through a mechanism called ribosomal frame shifting, or sometimes read-through. Basically, the ribosome slips or reads past the stop signal.
Why do that?
This allows the host ribosome to continue translating past the stop codon, producing a longer fused GAG-POL polyprotein, which contains both the GAG structural proteins and the essential POL enzymes, reverse transcriptase, integrase, protease. This ensures that the crucial pull components are produced, but typically in a regulated, usually lower amount compared to the more abundant structural GAG proteins needed for the virus particle itself.
Very clever. So after these proteins are made and new viral RNA is packaged, how do the new viruses get out?
New virions are assembled near the cell surface and then released from the cell through that process we mentioned earlier called budding from the cell membrane. During this budding process, host cell membrane proteins are frequently incorporated into the viral envelope alongside the viral glycoproteins.
Ah, that camouflage again?
Exactly. It can give the newly formed virions a deceptive appearance, making them look somewhat like the host cell, which might help them evade immediate recognition by the host immune system.
That's a truly sophisticated evasion tactic built right into their exit strategy. Now, you mentioned something earlier about the cellular requirements for proviral integration. Does this mean that cells that aren't actively dividing are generally resistant to retroviral infection?
In general, yes, that's correct. Proviral integration typically occurs during the S phase of the cell cycle. That's the phase when the host cell is actively synthesizing its DNA in preparation for cell division.
Okay.
This means that for most retroviruses, quiescent or non-dividing cells are largely resistant to infection because they aren't in the S phase, and the nuclear membrane might be intact, blocking access for the provirus. This is a significant limitation for many retroviruses, as many important target cells in the body aren't actively dividing all the time.
But you said most retroviruses. Is there an exception?
However, and here's a key and clinically very significant exception, only the lentiviruses, the subfamily that includes HIV, have evolved the unique ability to actively transport their pre-integration complex into the nucleus and infect non-dividing cells.
Ah, that's why HIV can infect cells like macrophages.
Precisely. This is one of the reasons lentiviruses like HIV are so formidable. They can establish persistent infections in long-lived non-dividing cells like macrophages and resting T cells, creating stable reservoirs in the body. And remember, once a host cell is infected by a retrovirus and that provirus integrates, it remains infected for the entire lifespan of that cell. This permanence is a crucial characteristic that contributes to the chronic, persistent nature of these infections.
That long-term infection aspect is truly profound. The cell basically becomes a permanent, potentially self-replicating viral factory.
Okay, let's decode the retroviral genome itself now. Looking at figures 2012 and 2013 in our source material, how is their genetic material organized? What are the key regions we need to understand? Retroviral genomes are remarkably compact and efficiently organized. They contain both coding regions, which provide the instructions for making the viral proteins, and non-coding sequences.
And the non-coding parts are important too.
Absolutely critical. The non-coding sequences don't produce proteins themselves, but they serve as essential recognition signals or regulatory elements for processes like DNA synthesis, RNA synthesis, and processing events. These crucial non-coding regions are typically found clustered in the 5-RNO and 3-RNL terminal regions of the genome. All retroviral genomes are characterized by being terminally redundant. This means they have identical sequences repeated at both ends, and these are called long terminal repeats, or LTRs.
LTRs. And these aren't just simple repeats, right? They sound like they're regulatory powerhouses.
They truly are. While the ends of the initial retroviral RNA genome differ slightly in sequence from the integrated retroviral DNA, the provirus, in that proviral DNA form, the LTRs are identical and are repeated at both the 5-RL and 3-RNA termini. These LTRs are indeed powerhouses of genetic control. They contain crucial regulatory sequences that are involved in initiating viral protein expression. They act as strong promoters and enhancers for viral gene transcription by the host machinery.
Okay.
They also contain signals important for facilitating the integration of the provirus into the host genome. And they control the polyadenylation of the viral RNAs, which is important for RNA stability and efficient translation. Additionally, other important non-coding sequences called viral packaging sequences are located just outside the LTR sequences. These are vital recognition sites that ensure the viral RNA genome gets correctly identified and packaged into new assembling virions.
So the LTRs are like the master control switches and integration guides. Then we get to the core structural genes, GAG, POL, and NVI. These must be the fundamental building blocks, common across most retroviruses. Let's break down the GAG gene first. What does it do?
Right, the GAG gene, it stands for group-specific antigen, or sometimes core protein, is essential for forming the internal structure of the virus particle. It encodes a single large precursor polyprotein.
A polyprotein.
Yeah, think of it as a long, continuous protein chain that actually contains multiple distinct functional proteins linked end-to-end. This large GAG precursor polyprotein is then subsequently cleaved or cut by a viral protease, into 3 to 5 smaller functional capsid proteins.
What are those smaller proteins?
They include the matrix MA protein, which lines the inner surface of the viral envelope, the capsid CA protein, and here's a fascinating detail of viral efficiency. A fraction of the GAG precursor polyproteins themselves also contain the viral protease enzyme embedded within them. This protease is responsible for cleverly cleaving both the GAG and the GAG-POL polyproteins into their smaller functional components during the viral maturation process, often after the virus is budded from the cell.
Wow, so it carries its own processing tool right within the structural protein. That's efficient. And POL, I imagine that's where the famous reverse transcriptase comes from.
Correct. The POL gene, standing for polymerase, specifically RNA-dependent DNA polymerase, is arguably the most crucial gene for the defining feature of retroviral replication. It encodes 3 absolutely vital enzymes. First, it encodes reverse transcriptase, RT, the very enzyme that copies the viral RNA genome into the double-strand DNA provirus, effectively inverting that central dogma. Second, it encodes integrase, IN, the enzyme that facilitates the precise and permanent insertion of that newly synthesized provirus into the host cell DNA. This is seen, for example, very clearly in HIV-1. And third, it encodes another crucial enzyme, the protease, PR. This is the enzyme specifically responsible for cleaving the GAG and GAG-POL polyproteins into their smaller functional protein products during the final stages of viral assembly and maturation.
So POL is truly the engine room for the retrovirus's genetic manipulation, replication, and protein processing, like a Swiss Army knife for the virus.
That's a great analogy. It handles everything from transcription and integration to key protein processing.
And the third core gene, ANV, that must be about the viral envelope, the part that interacts with the host cell.
Precisely. The ND gene, short for envelope, is responsible for producing the viral envelope glycoproteins. These are the proteins that get embedded in that outer lipid envelope the virus acquires from the host cell. They are absolutely critical for the virus's initial interaction with the host cell.
How so?
These glycoproteins typically include 2 main components that work together. There's the surface protein, SU, which is the outer component that specifically binds to those cell surface receptors on target host cells. This binding determines which cell types the virus can infect. That specificity is what we call viral tropism. And then there's the transmembrane protein, TM, which is a smaller protein that spans the viral envelope and essentially anchors the SU complex to it, playing a role in the fusion process that lets the virus core enter the cell. These envelope proteins are essential for both viral entry and for dictating the range of host cells the virus can successfully target.
Okay, so those 3, GAG, POL, and NV, are the core structural and enzymatic genes, pretty much common across all retroviruses. But then you get into the more complex human retroviruses, like HTLVs and lentiviruses like HIV, which have additional accessory genes. This is where their unique complexities and their advanced ability to manipulate host cells really come into play, isn't it?
This is absolutely where the sophistication truly ramps up. It's what distinguishes the relatively simple retroviruses from the complex, often highly pathogenic human ones. Human retroviruses, unlike simpler animal retroviruses, which might rely solely on GAG, POL, and NV, possess these additional regulatory or accessory genes.
Where are they located?
These accessory genes are typically located in the regions between the NV and the 3'-FTR, or sometimes overlapping other genes. And they are absolutely critical for the pathogenesis of these viruses, for their ability to replicate efficiently, and for their complex life cycles. They enable much more nuanced and intricate interactions with the host cell's internal machinery, its signaling pathways, and its immune responses.
Let's start by exploring the key accessory genes of the HTLVs, specifically HTLV-1 and HTLV-2. What are the most important ones there that we need to know about?
Okay, HTLVs have a significant region nestled between their NV gene and the 3'-LTR. This region encodes several important proteins and transcripts, often using overlapping reading frames, which is a way to pack more information into a limited genetic space.
Very efficient.
Indeed. The most prominent and well-studied of these HTLV accessory proteins is TAX. It's a 40-kilodate protein. Now, crucially, TAX does not bind directly to DNA itself, which is quite unusual for a protein that has such a profound effect on gene expression.
So how does it work then?
Instead of binding DNA, it acts as a powerful transactivator. It indirectly induces the expression of a wide range of host cell transcription factors. Think of it as influencing the influencers. These include members of the really important C-rel nuclear factor E and FK family, which controls inflammation and immunity, as well as ETS1 and ICA2, and members of the phosgen family involved in cell growth. This remarkable ability allows TAX to dramatically reprogram the host cell's gene expression profile.
So TAX acts like a master conductor for the host cell's genetic orchestra, even without directly touching the musical score itself.
That's a fantastic analogy, yes. It doesn't directly bind the DNA notes, but it pulls the levers that control the cell's own genetic switches. And its influence goes beyond just transcription factors. It also promotes the expression of various cytokine signaling molecules like interleukin-2, IL-2, granulocyte macrophage colony stimulating factor, GM-CSF, and tumor necrosis factor, TNF, which are key players in the immune system and cell growth.
And other things too.
Yes. Furthermore, it induces membrane proteins and receptors, like major histocompatibility, MHC molecules, involved in antigen presentation, and the IL-2 receptor 8 chain, important for T-cell growth. It even affects chromatin remodeling complexes, which can physically alter the accessibility of DNA for transcription. Here's a key insight for pathogenesis. TAX is capable of inducing cell transformation, pushing cells towards cancer under certain circumstances. And it's thought to be absolutely critical to the early phases of this transformation process in HTLV-1 infection, driving those initial steps towards malignancy.
And what about the REX protein in HTLVs? What role does that play?
REX, which is a 27-kilolide protein, plays a vital post-transcriptional role. So after the RNA is made, it regulates the expression of viral mRNAs. Its primary function is to facilitate the nuclear export of unspliced or singly spliced viral RNAs from the nucleus out into the cytoplasm.
Well, why is that important?
This is crucial for the production of the structural proteins, like GAG and POL, which are made from unspliced RNA, and for ensuring the full-length viral genome RNA gets packaged into new variants. Without REX, these essential RNAs would remain trapped in the nucleus or be improperly processed by the host cell splicing machinery.
So TAX controls transcription, REX controls RNA export. Are there others?
The HTLVs also produce some other transcripts, like P12, P13, and P30. These tend to have functions that restrict the overall expression of viral genes and seem to diminish the immunogenicity of infected cells, basically acting as a kind of viral immune evasion mechanism, helping the infected cell fly under the radar of the host's immune system.
And then there's HBZ, which sounds particularly intriguing because you mentioned it's transcribed from the opposite DNA strand. What's its function?
HBZ, or HTLV-1 basic leucine zipper domain-containing protein, is indeed fascinating. As you noted, it's a product of the complementary or antisense proviral DNA strand, meaning it's transcribed in the opposite direction compared to all the other viral genes like GAG, POL, NV, TAX, and REX.
What does it do?
It interacts with many different cellular transcription factors and signaling proteins. Its primary effect appears to be stimulating the proliferation, the growth, of infected T cells. And this is an extremely important detail for understanding HTLV-1 pathogenesis, particularly ATL. HBZ is the only viral product that is found to be universally expressed in virtually all HTLV-1 infected tumor cells in vivo.
The only one consistently expressed in the actual tumors.
Yes. This strong and consistent expression strongly suggests it plays an essential role in maintaining the transformed cancerous state, especially given the irregular or often completely absent expression of TAX in the later stages of malignancy. It's like TAX might start the fire, but HBZ keeps the embers glowing.
So HTLVs have their own complex set of regulatory tools to control their replication and impact the host cell. Now let's turn to the lentiviruses like HIV-1 and HIV-2. They have even larger genomes and more accessory genes, correct? What makes them so particularly complex and, well, insidious?
That's right. Lentiviruses, including HIV-1 and HIV-2, have a larger and significantly more complex genome compared to other pathogenic retroviruses like the HTLVs. They contain an untranslated region located between the POL and N genes that encodes portions of several distinct accessory proteins, often utilizing those same clever overlapping reading frames to maximize the genetic information packed into their relatively small genome.
And these accessory genes are key to their success.
Absolutely. These accessory genes are a testament to their sophisticated evolutionary strategies for manipulating host cell machinery, effectively evading diverse immune responses, and ensuring highly efficient replication. They contribute significantly to their ability to cause chronic, persistent, and ultimately severe disease like AIDS.
Okay, let's quickly run through these key lentiviral accessory genes. We have TAT and REV, which sound a bit like the HTLV ones, and then NEF, VIF, VPR, and VPU, or VPX in HIV-2. What does TAT do in HIV?
TAT, which is a small p14 protein in HIV, acts as an extremely potent transactivator of viral transcription. It dramatically boosts viral gene expression from the LTR promoter region. Think of it as a powerful supercharger for viral gene transcription.
How much does it boost it?
Orders of magnitude. It rapidly increases the efficiency of the host's RNA polymerase second enzyme on the viral template. ensuring the very high levels of viral RNA are produced very quickly. Without TAT, HIV replication is extremely sluggish and inefficient. Then there's REV, a P19 protein. It serves a role very similar to the REX protein of HTLV. REV plays a vital post-transcriptional role, regulating RNA splicing and GONDO-RNA transport. It's crucial for directing the appropriate processing and selective export of different viral messenger RNAs from the nucleus out to the cytoplasm. This ensures the correct viral proteins like GAG, POL, NV are produced at the right time and in the right amounts for efficient viral assembly.
Okay, TAT boosts transcription, REV controls RNA export. What about NEF? I've heard that one mentioned a lot in the context of HIV and its impact on the immune system.
Yes. NEF, a P27 protein, is a truly multifunctional accessory protein, and it is absolutely vital for efficient HIV pathogenesis in vivo. It performs several critical roles that help the virus thrive.
Like what specifically?
Notably, it downregulates the expression of CD4, which is the primary cellular receptor for HIV, on the surface of infected cells. This serves multiple purposes. It prevents superinfection of the same cell by other HIV particles, and it helps the infected cell evade recognition by certain immune cells.
Clever. What else?
It also ingeniously alters host T-cell activation signaling pathways, potentially modulating the host immune response in ways that actually benefit the virus's survival and spread. Furthermore, NEF significantly enhances the intrinsic infectivity of newly produced virions, making each individual virus particle more capable of successfully establishing infection in a new target cell.
So it's playing both defense for the infected cell by hiding it and offense for new variants by making them more potent. What about VIF?
VIF, a P23 protein, is necessary for the proper assembly of the HIV nucleoprotein core in many cell types, particularly primary T cells and macrophages. Without VIF, the viral core structure is often defective. This leads to inefficient production of proviral DNA in newly infected cells, severely hindering viral replication in these key target cells.
Okay, so important for assembly. Anything else?
Yes. Furthermore, VIF has a critical and direct role in combating a powerful host antiviral defense system. It specifically targets a host protein called ApoBec3G and related ApoBec proteins.
What does ApoBec do?
ApoBec proteins are part of our innate immune system. They are cytidine deaminases, meaning they normally get incorporated into the viral DNA during reverse transcription and mutate it by changing cytosine bases to uracil. This hypermutation essentially inactivates the viral genome.
So a host defense mechanism.
A potent one. But VIF efficiently targets these ApoBec proteins for destruction by the cell's own proteasomal degradation machinery. By destroying ApoBec, VIF effectively blocks its antiviral effect and allows the virus to replicate unhindered. It's a fascinating example of the ongoing molecular arms race between virus and host.
Wow, actively destroying our defenses. That sounds like a sophisticated counter-strategy.
Right.
And VPR, I recall you mentioned it's key to lentivirus's unique ability to infect those non-dividing cells.
Yes. VPR, a small P15P16 protein, is a key factor behind why lentiviruses, uniquely among known retroviruses, have evolved the ability to efficiently infect non-dividing cells.
How does it achieve that?
VPR facilitates the active transport of the viral pre-integration complex, that's the proviral DNA, along with associated viral proteins like integrase through the nuclear pore complex into the nucleus. This circumvents the need for the nuclear envelope to break down during mitosis, which is something other retroviruses generally require for the DNA to access the host chromosomes.
So it unlocks the door to the nucleus in resting cells.
Exactly. This means HIV can infect vital, long-lived cells that don't regularly divide, like resting memory T cells and macrophages, establishing those persistent reservoirs. Additionally, VPR can induce other cellular changes, such as causing a temporary halt in the cell cycle of the G2 phase, G2 arrest, in infected cells, and it can also induce differentiation of some target cells, like macrophages. These effects might create a more favorable and stable environment for viral replication and long-term persistence.
And finally, V2 and VPX. What do they contribute to the lentiviral strategy?
Okay, VPR, a P16 protein, is specific to HIV-1. It's not found in HIV-2. It has two main well-characterized functions. First, it promotes the degradation of newly synthesized CD4 molecules in the endoplasmic reticulum, preventing them from reaching the cell surface. This helps ensure efficient viral budding and also prevents CD4 from interfering with the envelope proteins.
And a second function.
Second, VP stimulates the release of new virions from infected cells by counteracting a host restriction factor called tetherin, also known as BST2. Tetherin normally acts like molecular glue, physically trapping newly formed virions on the cell surface and preventing their release. VPU effectively neutralizes tetherin, allowing the virus particles to escape efficiently.
Another counter-defense mechanism. And VPX.
VPX is an accessory protein that's found in HIV-2 and also in many related simian immunodeficiency viruses, SIVs, but not HIV-1. It's structurally related to VPR. While its specific functions in HIV-2 pathogenesis are still being fully defined, it's known to play a crucial role, similar in concept to VIF, in counteracting another host restriction factor called SAMHD1, particularly in myeloid cells like macrophages and dendritic cells. By degrading SAMHD1, VPX helps ensure efficient reverse transcription in these otherwise restrictive cell types. So these accessory genes, taken together, really highlight the incredible complexity, the adaptability, and the sophisticated strategies that human retroviruses, especially lentiviruses, have evolved to establish persistent infections and ultimately cause disease.
That's an absolutely astonishing level of detail and control packed into such tiny entities. It truly shows how incredibly sophisticated these viruses are in their interactions with our own cells. Okay, now that we have a much better handle on their structure and their really intricate life cycle, let's explore retroviral pathogenesis, how they actually cause disease, and also touch on some of their emerging applications, particularly in gene therapy. Starting with the spectrum of retroviral pathogenicity, how do those basic core genes, GAG, POL, and ENVA, relate to a retrovirus's ability to cause disease on their own?
It's a general observation that retroviruses contain only those basic GAG, POL, and M genes, the ones often referred to as simple retroviruses, typically either are not significantly pathogenic in their natural hosts, or if they do cause disease, it often takes an extremely long time, maybe even the host's entire lifespan, to induce that disease.
So the core genes alone usually aren't enough for severe disease.
Generally not for rapid or severe disease, no. This observation really highlights the critical importance of those other regulatory genes, the accessory genes, that we just spent time dissecting in the more complex retroviruses. It is these additional accessory genes that are truly responsible for the more acute or severe viral disease pathogenesis seen in human retroviruses like HIV and HTLV-1. Without them, the viruses are often much less virulent, much less aggressive.
That makes perfect sense. The accessories aren't just optional extras, they're essential drivers for significant disease. Okay, now let's talk about one of the most profound and frankly concerning aspects of retroviruses, their mechanism of neoplastic transformation. How exactly can they cause cancer?
Right, the pathogenesis of neoplastic transformation by retroviruses turning a normal cell into a cancerous one often relies on one of two main mechanisms. The first mechanism involves what's essentially chance integration of the provirus at a very specific, unfortunate spot in the host genome.
Unfortunate for the host, that is.
It's exactly. This is called insertional mutagenesis. While the integration site itself is largely random for human retroviruses, if the provirus happens, just by chance, to integrate near a cellular gene that normally regulates cell growth, a gene known as a proteogene gene, its powerful LTR promoter enhancer sequences can inappropriately switch on or dramatically overactivate the expression of that nearby proteogene.
Turning it into an oncogene.
Precisely, turning it into a transforming oncogene. This leads to unregulated cell proliferation and can ultimately drive the development of cancer. A classic example from animal studies is the avian leukosis virus. It causes B-cell leukemia in chickens, specifically by inducing the overexpression of the host's mycocogene when the provirus integrates adjacent to it. The integration event itself isn't inherently oncogenic, but its location can be catastrophic by disrupting normal gene regulation.
So it's like a random but potentially devastating genetic hit based purely on location. But you also mentioned that some retroviruses actually carry oncogenes within their own viral genome, don't they?
Yes, that's the second major mechanism, and it represents a slightly different, often more aggressive class of retroviruses, mostly found in animals. Some retroviruses possess cellular genes that they have literally captured from a previous host genome during their evolutionary history and incorporated directly into their own viral genome.
They steal host genes.
Essentially, yes, over evolutionary time. These captured and often mutated or altered cellular genes are then called viral oncogenes, or VONCs. And because the virus now carries this potent cancer-causing gene, it can directly and often very rapidly transform the infected host cell into a cancerous one upon infection. These are known as acute transforming retroviruses.
They sound dangerous. Are they common?
Well, here's a key point about them. Viruses that carry such powerful oncogenes often have had to delete a portion of their own essential viral genome, like parts of GAG, POL, or ENVI, to make space for the oncogene. This means that such viruses, while acutely transforming, are often replication defective. They can transform a cell, but they can't produce new infectious virus particles on their own. They require helper viruses, other non-oncogenic retroviruses, infecting the same cell to provide the missing viral proteins needed for them to reproduce and spread.
Ah, so they need assistance.
Yes. This dependence on helper viruses might explain why these acute transforming retroviruses, while potent, are actually quite rare in nature and particularly seem to be absent in human populations studied so far.
And crucially, what about the known human retroviruses in this context? Do HIV or HTLV carry their own viral oncogenes, making them acutely transforming?
This is a really important distinction. Importantly, all human retroviruses identified to date, like HIV-1, HIV-2, HTLV-1, and HTLV-2, are exogenous, meaning they are acquired by infection from external sources. They aren't inherited as part of our normal germline DNA. And critically, they are not acutely transforming viruses. Meaning they do not possess an intrinsic, direct transforming viral oncogene within their own genome that would rapidly induce cancer immediately upon infection. Their oncogenic potential, like we see clearly with HTLV-1 causing APL, arises through much more indirect and often very long-term mechanisms. These involve the complex actions of their accessory proteins like TAX and HBZ altering host cell pathways over time, or potentially through that slower process of insertional mutagenesis we discussed, rather than them carrying a dedicated, rapidly cancer-causing gene themselves.
That's a really important distinction, setting them apart from those acutely transforming animal retroviruses we hear about. Okay, now, despite their obvious disease-causing potential, the remarkable properties of retroviruses, particularly their innate ability to integrate their genetic material permanently into the host genome, have led to extensive experimental efforts to actually use them as therapeutic tools, as vectors in gene therapy or gene transfer. It sounds like an incredibly powerful concept, but also, perhaps, a double-edged sword.
It absolutely is both of those things. Their unique ability to stably and permanently integrate new genetic material into host cells makes them incredibly appealing, conceptually, as gene therapy vectors. They could serve as biological delivery vehicles to introduce new genes into target cells.
What kind of potential applications are we talking about?
Well, the therapeutic potential is vast, at least in theory. We could potentially use them to repair a genetic defect, for example, by delivering a functional copy of a faulty gene to a patient with an inherited genetic disorder, like certain immunodeficiencies or metabolic diseases. Or, conversely, we could introduce an entirely new gene that confers a new therapeutic property to the cell. A classic conceptual example often cited is inserting a suicide gene, like semidine kinase, specifically into tumor cells. This would make only the tumor cells uniquely susceptible to being killed by a specific, otherwise relatively harmless drug, like ganciclovir, allowing for targeted destruction of cancer cells while sparing normal tissues.
That's an incredible vision, turning a pathogen's core mechanism into a potential therapeutic tool. But you mentioned a double-edged sword. What are the significant concerns or risks associated with using retroviral vectors in humans, given their inherent nature?
Despite the immense promise and decades of ongoing research, there's a significant and very well-founded source of concern regarding the widespread clinical use of retroviral vectors in humans. One major risk is the potential for insertional mutagenesis, similar to how they can cause cancer naturally. If the therapeutic vector integrates into the wrong place in the host genome, it could potentially activate a proto-oncogene or inactivate a vital tumor suppressor gene, inadvertently leading to cancer in the patient being treated. This has actually happened in some early gene therapy trials, sadly.
So the integration site is a major safety hurdle.
A huge one. Another concern is the risk that replication-competent viruses might inadvertently be generated during the vector production process, or perhaps even more subtly, that the introduction of the vector could somehow rescue or activate endogenous retroviral sequences.
Endogenous retroviruses, what are those?
These are ancient viral sequences that integrated into the genomes of our ancestors millions of years ago and became part of our heritable human germline DNA. Most are now degraded and non-functional, essentially molecular fossils, but they still exist within our DNA, making up a surprising percentage of it. The concern is that the introduction of new active viral components via gene therapy could lead to unpredictable results, potentially reactivating some of these dormant viral elements or leading to recombination events.
Is that just a theoretical worry?
Well, this concern isn't merely hypothetical. The fact that proteins encoded by some of these endogenous retroviral sequences have actually been detected on the surface of certain human cancer cells implies that the genetic events leading to that cancer were somehow able to activate the synthesis of these usually silent ancient viral genes. This is a powerful so what moment for you, the listener, because it underscores the profound real-world implications and complexities. Harnessing the power of retroviruses for therapy means not only understanding their potential benefits, but also rigorously mitigating the inherent risks of inserting foreign genetic material, especially when it involves elements derived from viruses that have such a deep intertwined history with our own genome.
That is truly fascinating, and as you said, definitely a bit unnerving at the same time. The idea that we carry silent, ancient viral genes within us that could potentially be reactivated, it certainly underscores the depth of knowledge and the extreme caution required in this field. Okay, let's shift now to a much more detailed look at the specific human T-cell lymphotropic viruses, or HTLVs. Let's start with HTLV-1, a delta retrovirus, and its associated diseases. How was it first discovered, and what were the initial observations back then?
Right, HTLV-1 holds a really significant place in the history of virology and cancer research. It was first isolated back in 1980. It came from a T-cell lymphoma cell line that was derived from a patient in the U.S. Initially, it was thought that this patient had a known form of skin lymphoma called cutaneous T-cell lymphoma.
And it was.
But it later became clear that the patient actually had a distinct and at that time largely unrecognized form of T-cell malignancy. This distinct form had actually been previously reported, mainly in Japan, and was eventually named Adult T-cell Leukaemia lymphoma, or ATL. So the virus discovery went hand in hand with defining a new disease entity. And this discovery was a true landmark, as HTLV-1 became the very first human retrovirus definitively linked to a human cancer.
So a new virus identified alongside a new specific type of cancer. And fast forwarding to today, what are its key well-established disease associations based on decades of serologic data and epidemiological studies?
Through extensive research over the following decades, HTLV-1 has been definitively linked to two major distinct clinical diseases. The first is, as mentioned, Adult T-cell Leukemia Lymphoma, ATL, which is that distinct and often aggressive malignancy of mature T lymphocytes. The second major disease is Tropical Spastic Paraparesis, TSP, which is also known as HTLV-1-Associated Myelopathy, HTMPO. This is a chronic progressive neurological disorder primarily affecting the spinal cord.
So a cancer and a neurological disease. Any others suspected?
Beyond these two primary associations, HTLV-1 is also suspected to play a role in, or at least contribute to, other conditions. These are often referred to as putative associated conditions and include things like infective dermatitis, a chronic skin condition, certain forms of arthritis, uveitis, which is an inflammatory eye condition, and perhaps Sjogren's syndrome, an autoimmune disorder affecting glands. However, it's important to stress that the causal links for these latter conditions require further definitive confirmation. The evidence isn't as strong as it is for ATL and HAVs.
Okay, understood. Now let's take that deeper dive into the biology and molecular biology of HTLV-1 itself. What do we know about its cellular receptors? How does it actually get into our cells? And how does it establish that infection, particularly regarding the concept of latency?
Okay, so HTLV-1 uses a specific set of host proteins on the cell surface to gain entry. The human glucose transporter protein 1, known as GLUT1, has been identified as functioning as a primary receptor for HTLV-1.
The glucose transporter. That's interesting.
It is, and it likely works in concert with other co-receptors on the cell surface, such as neuropilin-1, NRP1, and also heparan sulfate proteoglycans, to facilitate efficient viral binding and entry into the target T cell. Now, what's particularly interesting and really key to understanding its long-term behavior in vivo, meaning within a living person, is that the most common outcome of HTLV-1 infection is actually latent carriage.
Latent carriage?
Yes, the randomly integrated provirus just sits there, predominantly within CD4 plus T cells, but it's transcriptionally silent. Crucially, in the vast majority of these latently infected cells, there's no active viral gene product expression. The virus is essentially dormant, hiding from the immune system. This is a key distinguishing feature. And remember what we said earlier, the only viral gene product that is consistently found expressed in the actual tumor cells transformed by HTLV-1 in vivo is HBA.
So it can just sit there silently for years, potentially decades, without making any viral proteins that the immune system could detect.
That's quite a stealth strategy.
And what about tax's role in transformation? You mentioned it's thought to be critical for initiating the process, but then it's often not expressed in the later stages of the cancer cells. How does that apparent paradox work?
That is one of the most profound and somewhat puzzling aspects of HTLV-1 pathogenesis. The tax gene, as we discussed, is thought to be absolutely critical to the initiation of the cell transformation process. It provides that initial push towards uncontrolled growth by activating all those host genes.
Right.
However, in a surprising twist, TAX protein is often not expressed, or its expression is significantly downregulated, in the actual tumor cells found in many ATL patients, especially in the more aggressive forms of the disease.
Why would that happen if it's so important for transformation?
Well, the leading hypothesis for this lack of expression in established tumors is that TAX-expressing cells are highly immunogenic. TAX itself and the cellular genes it activates make the cell look very abnormal to the host's immune system, particularly cytotoxic T lymphocytes. So these TAX-expressing cells are likely recognized and eliminated by the host immune system over time. Therefore, there's strong selective pressure against TAX expression in vivo as the clone evolves toward full malignancy. Cells that manage to shut down TAX expression might survive better.
So the immune system selects for cells that hide the TAX protein.
That seems to be the case. And this contrasts sharply with what happens in vitro. Cells transformed by HTLV-1 in a laboratory dish typically do actively transcribe HTLV-1 RNA and produce infectious virions, including TAX. This difference makes it quite difficult to establish stable long-term cell lines directly from authentic ATL patient tumor samples. It really highlights the dynamic interplay between viral gene expression strategy and host immune pressure within a living organism.
That's a crucial distinction between lab results and the real-world situation in a patient. It's like the virus changes its strategy to survive long-term, and even though TAX doesn't bind DNA directly, you mentioned it dramatically alters host gene expression. How does it achieve such a widespread impact on cellular functions, pulling all those levers?
Indeed. Even without direct DNA binding, the TAX protein is primarily located in the nucleus, and it exerts this dramatic and widespread influence on host gene expression. It functions as that powerful molecular switch, indirectly inducing the expression of a vast array of host cell gene products. This includes those key transcription factors, particularly in the NFE family, but also its 1, its 2, and members of the phosgene family, all critical regulators of cellular processes.
Right.
And beyond transcription factors, it actively promotes the expression of various cytokines, those signaling molecules like IL-2, GM-CSF, and TNF, which are critical for cell growth, inflammation, and immune responses. It also induces membrane proteins and receptors like MHC molecules and the IL-2 receptor I chain, and it even affects chromatin remodeling complexes, which physically influence how genes are packaged and accessed for transcription. The genes that get activated by TAX are generally known to be controlled by transcription factors of the CRO NFB and the CR protein families, demonstrating its sophisticated hijacking of these core cellular signaling pathways to promote cell survival and proliferation.
So it's a central orchestrator of the cell's internal environment, pushing it towards a pro-growth, pro-survival state. But if ARIS isn't consistently expressed in ATL cells in the long run, how does HTLV-1 infection ultimately lead to full-blown cancer? And what role, again, does HBZ play in maintaining that malignant state if TAX fades away?
This remains one of the central, enduring puzzles in understanding HTLV-1 pathogenesis. While TAX's initial transforming effects are quite clear in experimental systems, it can interfere with critical cell cycle checkpoints, like G1 and mitotic checkpoints. It can block apoptosis, program cell death, it can inhibit DNA repair mechanisms, and it can promote antigen-independent T-cell proliferation, the precise molecular mechanism by which this widespread induction of host gene expression ultimately leads to full, stable neoplastic transformation in vivo is still not entirely clear.
Any leading theories?
Well, the idea of inducing a cytokine autocrine loop has been proposed. This is where the transformed cells perpetually stimulate their own growth by secreting and responding to their own growth factors. Although, interestingly, IL-2 itself, which TAX induces, doesn't seem to be the crucial cytokine for maintaining ATL growth. Instead, other cytokines like IL-4, IL-7, and particularly IL-15 have been suggested as potentially more important players in sustaining the tumor.
And HBZ's role in maintenance?
Given the inconsistent or often absent expression of TAX in established ATL tumor cells in vivo, the HBZ gene product, transcribed from that antisense strand, is increasingly thought to be essential for the maintenance of the transformed state. Its universal presence in tumor cells strongly suggests it provides the continuous proliferative signals needed for the cancer cells to persist and expand, perhaps taking over that role from TAX.
So maybe TAX initiates the process, gets things rolling, maybe causing some genetic instability, and then HBZ takes over to keep the transformed cells proliferating long-term, possibly while being less visible to the immune system than TAX.
That's a very plausible model, yes. TAX kicks off the cancerous process, perhaps contributing to initial genetic damage, and HBZ seems crucial for keeping it going, for maintaining that malignant state.
And it seems that HTLV-1 infection only very rarely leads to full-blown cell transformation in most infected people. This strongly suggests a multiple hit hypothesis, where other genetic events beyond just the virus are needed for cancer to develop. Is that the current thinking?
Exactly. This rarity of transformation, which is very clearly seen in the epidemiology of HTLV-1, remember only about 3% lifetime risk of ATL, strongly supports a multiple hit hypothesis. It suggests that the progression from a latently infected T cell to a fully malignant ATL cell is a rare event, likely depending on the accumulation of additional heterogeneous second, third, or even fourth genetic hits within the host cell's own genome, occurring on top of the initial viral infection and the effects of TAX and HPZ.
Are there specific genetic hits commonly found?
While no single consistent chromosomal abnormality has been described across all ATL cases, unlike some other leukemias, aneuploidy, having an abnormal number of chromosomes, is very common, suggesting widespread genomic instability. Specific mutations in important genes like the p53 tumor suppressor gene and certain T-cell receptor gene translocations have been reported in individual cases, but there's no single smoking gun found in everyone. It's hypothesized that TAX's known ability to repress certain DNA repair enzymes might permit the accumulation of this diverse genetic damage over the many years of chronic infection, effectively lowering the threshold for these subsequent hits to occur. However, despite significant research, the precise molecular pathogenesis of HTLV-1-induced neoplasia is still not fully understood. It remains a complex interplay of viral factors, HBZ, host genetics, accumulated mutations, and likely chance events over a long period.
That makes it a particularly challenging puzzle for researchers and clinicians to fully unravel. Okay, let's move on to the features of HTLV-1 infection from a broader perspective, specifically its epidemiology. Understanding how it spreads is crucial. How is HTLV-1 transmitted between humans?
HTLV-1 infection is transmitted in at least three key and well-established ways. The first, and arguably the most important route for determining long-term disease risk, particularly for ATL, is vertical transmission from mother to child. This occurs especially via infected lymphocytes in breast milk. This is recognized as the most common route for perinatal transmission, especially in highly endemic regions.
Okay, mother to child via breastfeeding. What else?
The second major route is through sexual activity. Importantly, epidemiological studies show it's more commonly transmitted from infected men to women than vice versa, suggesting male to female transmission is more efficient. And the third significant route is through the blood. This can occur through transfusion of contaminated cellular blood products, like whole blood or packed red cells, less so with plasma, or through the sharing of contaminated needles and syringes among injection drug users, which parallels the transmission patterns seen with other bloodborne viruses like HIV and hepatitis C.
And how infectious is HTLV-1 compared to, say, HIV, which can also be transmitted via these routes? Is it easier or harder to transmit?
Compared to HIV, which can transmit quite efficiently even via cell-free viral particles found in bodily fluids, HTLV-1 is generally considered significantly less infectious per exposure event. Its transmission usually requires efficient cell-to-cell contact, meaning it's primarily the infected cells themselves, lymphocytes carrying the integrated provirus, rather than free-floating viral particles that are the main vectors of transmission. This implies that close, often prolonged contact involving the transfer of infected cells, as occurs in breastfeeding, sexual intercourse, or sharing blood-contaminated needles, is often necessary for successful transmission to occur.
That's an important distinction that definitely influences its epidemiology and spread patterns. What's its global distribution like? Is it found everywhere equally, or is it concentrated in specific endemic regions around the world?
HTLV-1 has a very distinct and somewhat patchy global distribution. It is highly endemic in certain specific areas. Most notably, southwestern Japan, including the islands of Kyushu, and particularly Okinawa, where historically over 1 million persons were estimated to be infected. Seroprevalence rates in these areas could be remarkably high, sometimes reaching up to 35% in certain older Okinawan populations, although rates are declining with prevention efforts.
Wow, 35% in some groups. That's incredible. Where else is it highly endemic?
Beyond Japan, significant infection clusters and high endemicity are noted in other parts of Eastern Asia, such as Taiwan. Also, throughout the Caribbean basin, including northeastern South America, like Colombia, French Guiana, also parts of northwestern South America. Central and southern Africa have significant endemic foci. And there are also distinct pockets of endemicity found within Italy, Israel, Iran, parts of Papua New Guinea, some indigenous populations in Australia and the Arctic, and even historically in the southeastern part of the United States, particularly among African Americans.
So, quite widespread geographically, but concentrated in specific hotspots. What's the estimated global burden?
Globally, it's currently estimated that somewhere between 5 and 10 million persons are living with HTLV-1 infection worldwide. The distribution map in Figure 201-4 in our source gives a good visual representation of these endemic areas.
That's a significant number of people worldwide. Yet you mentioned earlier that despite this high prevalence in certain areas, the actual incidence of disease, like ATL, seems remarkably low. Could you elaborate on that stark discrepancy?
Yes, that's one of the most striking paradoxes of HTLV-1 infection. And it ties back directly to that multiple hit hypothesis we discussed regarding cancer development. Despite the high prevalence rates in certain endemic areas, for instance in Japan, where historically over 1 million people were infected, only about 500 or so new cases of ATL are diagnosed there each year.
So a tiny fraction develop the cancer.
A very small fraction. This starkly indicates that clinical disease development is actually a rare outcome for the vast majority of individuals infected with HTLV-1. Most carriers will remain completely asymptomatic throughout their entire lives, highlighting that merely being infected with the virus is necessary, but clearly not sufficient for disease to occur. Other factors, those hits, or perhaps host genetic factors, are required.
So only a small percentage of infected individuals actually develop one of the associated diseases. What's the specific lifetime risk of developing either ATL or HAM for someone who is infected with HTLV-1? And what are the typical latency periods before these diseases appear?
Okay, based on long-term cohort studies, the estimated cumulative lifetime risk of developing ATL is approximately 3% among individuals infected with HTLV-1. Interestingly, there seems to be a consistent threefold greater risk among men compared to women for developing ATL, and the latency period between acquiring the initial infection, often in infancy, and the emergence of ATL is typically remarkably long, usually ranging from 20 to 30 years or even longer, highlighting the very chronic nature of the transformation process. ATL is also observed to be more common among individuals who are infected perinatally, mother to child, likely due to that prolonged duration of infection allowing more time for those secondary hits to accumulate.
Okay, so 3% risk for ATL, mostly in men, very long latency linked to perinatal infection. What about HAM, the neurological disease?
For HAM, the cumulative lifetime risk is projected to be roughly similar, perhaps around 4%. But in stark contrast to ATL, HAM more commonly affects women than men. The median latency period for HAM is generally shorter than for ATL, averaging around 3.3 years according to some studies. Although the range is extremely wide, it can vary from as short as 4 months after infection to as long as 30 years later.
And transmission routes.
Interestingly, transmission route seems to matter here too. While ATL is more linked to perinatal infection, a significant proportion, maybe around 20%, of patients who develop HAM acquired HTLV-1 from contaminated blood transfusions later in life. And HAM also appears to be somewhat more common among persons infected via sexual transmission compared to ATL. So different transmission routes might subtly influence the risk towards different disease outcomes, although the reasons aren't fully clear.
So different risks, different gender biases, different typical latencies, and maybe even different transmission routes predisposing to one disease over the other. Fascinating complexity. And just to confirm, do the geographic distributions of both ATL and HAM consistently overlap with those HTLV-1 endemic regions you mentioned, providing strong epidemiological support for the causal link?
Yes, absolutely. There's a very strong geographical correlation. Both the distributions of diagnosed ATL cases and HAM cases directly overlap with the known HTLV-1 endemic regions worldwide. And clinically, well over 95% of patients diagnosed with either typical ATL or HAM demonstrate clear serologic evidence of HTLV-1 infection, antibodies in their blood. This provides really robust epidemiological evidence supporting the causal link between this specific virus and these two distinct diseases.
Okay, let's delve further into the details of these associated diseases now, starting with Adult T-cell Leukemia Lymphoma, or ATL. What are its general defining characteristics, and how many distinct clinical types of this HTLV-1-induced neoplasia are generally recognized by clinicians?
Right, ATL. All ATL tumors are universally characterized as being monoclonal proliferations of mature post-thymic CD4 plus T lymphocytes.
Monoclonal, meaning?
Meaning that all the malignant cells within a patient originated from a single ancestral infected cell that underwent transformation. This is evidenced by demonstrating clonal proviral integration sites, meaning the virus is integrated into the exact same spot in the DNA genome of all the cancerous cells in that patient. They also exhibit clonal T-cell receptor gene rearrangements, further confirming their origin from a single T-cell clone. Clinically, ATL is generally classified into 4 distinct subtypes based on clinical features and prognosis. These are acute ATL, lymphomatous ATL, chronic ATL, and smoldering ATL. Each has quite unique clinical courses and expected outcomes.
Let's start with the most common one, acute ATL, which you said accounts for roughly 60% of all cases. What's its typical clinical course like, and what are its distinguishing hallmarks that clinicians look for?
Acute ATL is, as the name suggests, the most common and by far the most aggressive form of the disease. It's typically characterized by a very short clinical prodrome, often only about 2 weeks or so, between the patient noticing the first symptoms and getting a definitive diagnosis. And unfortunately, it has a very aggressive natural history, often with a devastatingly short median survival period of just around 6 months if left untreated, or if it doesn't respond well to therapy.
It's a very rapid progression. What are the common symptoms or signs?
The clinical picture is frequently dominated by several key features. Rapidly progressive and often widespread skin lesions, rashes, nodules, tumors, significant pulmonary involvement, infiltrates on chest x-rays, shortness of breath, severe hypercalcemia, that's abnormally high calcium levels in the blood, which can cause various symptoms, and usually a marked lymphocytosis, meaning an abnormally high number of lymphocytes, specifically the malignant T cells circulating in the peripheral blood. A classic diagnostic hallmark often seen on a blood smear is the presence of these distinctive malignant T cells, which have highly convoluted, indented, or multilobulated nuclei, sometimes described poetically as flower-shaped nuclei. These are quite characteristic morphologically, as illustrated in Figure 108-7 in the source material.
Flower-shaped nuclei, that's a very specific and memorable morphological clue for diagnosis. And what about the malignant cells themselves, beyond just their appearance under the microscope? What are their typical immunophenotypic markers, and are there other common clinical features or complications to be aware of in acute ATL?
Right, these malignant flower cells consistently show evidence of monoclonal proviral integration, confirming their origin. Immunophenotypically, they express specific markers on their surface. They are CD4 positive, like normal helper T cells, CD3 positive, a pan T cell marker, and notably, they strongly express CD25, which is the alpha chain of the IL-2 receptor. Interestingly, the soluble form of CD25 shed into the blood can sometimes serve as a useful tumor marker, its levels correlating with the overall tumor burden.
Okay. What about blood counts and bone involvement?
Unlike many other types of leukemia, significant anemia, low red blood cells, and thrombocytopenia, low platelet counts, are relatively rare, at least initially in ATL. The skin lesions, as mentioned, are common and can sometimes be challenging to distinguish morphologically just by looking at them from another type of T-cell skin lymphoma called mycosis fungoides, requiring biopsy for confirmation. A common but somewhat perplexing feature is the presence of lytic bone lesions areas where bone is being destroyed seen on X-rays. Crucially, these bone lesions typically do not contain tumor cells themselves. Instead, they seem to be composed of activated osteolytic cells, osteoclasts, which are bone-resorbing cells, usually without much accompanying osteoblastic activity, new bone formation. And despite the often high white blood cell counts in the blood, bone marrow involvement by the leukemia is frequently patchy rather than diffuse in most cases.
That hypercalcemia you mentioned sounds particularly concerning and potentially dangerous. What actually causes it in ATL patients? Is it the bone lesions?
The hypercalcemia seen in ATL is indeed a significant and often problematic complication, contributing to symptoms like confusion, constipation, and kidney problems. It appears to be multifactorial in origin. The tumor cells themselves are known to produce potent osteoclast activating factors, cytokines like tumor necrosis factor alpha, TNF-A, interleukin-1, IL-1, and lymphotoxin. These cytokines directly stimulate osteoclasts, the cells responsible for bone breakdown, leading to excessive calcium release from the bones into the bloodstream.
So the tumor cells tell the bone cells to dissolve bone.
Essentially, yes. And in addition to that, in some cases, the ATL tumor cells have also been shown to produce a parathyroid hormone-related protein, PTHrP, a molecule that mimics the action of parathyroid hormone, which normally regulates calcium homeostasis. Production of PTHrP can further contribute to the elevated calcium levels by increasing calcium resorption from bone and kidney tubules.
And you mentioned earlier that patients with ATL are also highly susceptible to opportunistic infections, similar to what's seen in AIDS patients. That suggests a significant underlying immunodeficiency is also part of the disease.
Yes, that's a critical and often life-threatening aspect of ATL, contributing significantly to morbidity and mortality. Affected patients do indeed have an underlying profound immunodeficiency state, even though their malignant cells are derived from T helper cells. This makes them highly susceptible to a wide range of opportunistic infections. Very similar in spectrum to those seen in patients with advanced AIDS, Although the precise pathogenesis of this immunodeficiency in ATL is still not fully clear, it's likely multifactorial, involving both dysfunction of the malignant clone and effects on normal immune cells.
What kind of opportunistic infections are common?
For example, pulmonary infiltrates seen on chest x-rays in ATL patients are often not due to the leukemia cells infiltrating the lungs, but are frequently opportunistic infections. Particularly common are infections with organisms like Pneumocystis jirovecii, formerly PCP, fungi like Candida or Aspergillus, viruses like cytomegalovirus, CMV, and also parasites, especially strongyloids, stercoralis.
Strongyloids, again, you mentioned an interesting link there.
Yes, this is an interesting and clinically significant connection. Strongyloids stercoralis is a parasitic roundworm found in the gut, and it happens to have an endemic distribution in tropical and subtropical regions that remarkably overlaps with many HTLV-1 endemic areas. There's a strong clinical observation and growing evidence suggesting that co-infection with strongyloids may not only be more common in HTLV-1 carriers, but might also potentially increase the risk of developing ATL or perhaps lead to its development more rapidly in those co-infected individuals. The parasite itself can cause hyperinfection syndromes in immunocompromised hosts.
That's a fascinating potential interplay between the virus, a parasite, and the host's immune system affecting cancer risk. Are there any other notable laboratory findings or patterns of CNS involvement that clinicians should be aware of in acute ATL?
In terms of general laboratory findings, serum concentrations of lactate dehydrogenase, LDH, are often significantly elevated, usually reflecting high tumor burden and rapid cell turnover. Alkaline phosphatase levels may also be elevated, particularly if there's significant bone or liver involvement. Regarding central nervous system, CNS involvement, it occurs in about 10% of acute ATL patients. Typically, it manifests as leptomeningeal involvement, meaning the malignant ATL cells infiltrate the membranes, meninges, surrounding the brain and spinal cord.
What symptoms does that cause?
This infiltration can lead to a variety of neurological symptoms, such as progressive weakness, altered mental status or confusion, paresthesias, abnormal sensations like tingling or numbness, and/or persistent headache. What's somewhat unique compared to many other CNS lymphomas is that ATL patients with CNS involvement can sometimes have normal cerebrospinal fluid, CSF protein levels, which might be misleading. Therefore, the definitive diagnosis relies on the careful cytological examination of the CSF to identify the characteristic malignant ATL flower cells.
Okay, that provides a very thorough picture of acute ATL. What about the other three clinical types you mentioned? Lymphomatous, chronic, and smoldering ATL. How do they differ in presentation and prognosis?
Right, the second recognized type is lymphomatous ATL. This accounts for about 20% of ATL cases. In terms of its natural history and generally aggressive clinical course, it's quite similar to the acute form. However, it's distinguished primarily by having rare or absent circulating abnormal flower cells in the peripheral blood, despite having widespread disease. Instead, the dominant feature is prominent lymphadenopathy, significantly swollen lymph nodes, often in multiple areas of the body, neck, armpits, groin. There might also be involvement of other organs like the liver or spleen. The histology, the microscopic appearance of the affected lymph nodes can be variable and doesn't necessarily predict the prognosis strongly. Diagnosis is usually suspected based on the patient's origin from an endemic region, perhaps characteristic skin lesions and maybe hypercalcemia, and it's definitively confirmed by finding serum HTLV-1 antibodies and demonstrating the malignant T-cell clone in the lymph node biopsy.
So similar aggressiveness to acute, but primarily presenting as lymphoma in the nodes rather than leukemia in the blood. What about the chronic form? How does that present, and what's its typical prognosis like?
Chronic ATL patients generally present with a much more indolent, slower-moving course compared to the acute or lymphomatous forms. They typically have normal serum calcium levels and normal or only slightly elevated lactate dehydrogenase LDH levels. Importantly, there's usually no significant involvement of the CNS, bone, or the gastrointestinal tract. Skin lesions and some lymph node enlargement might be present, and there might be a lower number of abnormal cells in the blood than in acute ATL. The median survival for patients presenting with chronic ATL is considerably longer than for acute ATL, averaging around 2 years from diagnosis. However, and this is a crucial point, it's known that in a significant proportion of cases, chronic ATL can eventually transform or progress into the more aggressive acute form, necessitating careful ongoing monitoring.
Okay, slower, but carries a risk of transforming into the acute phase. Finally, what about smoldering ATL? The name itself suggests it's the most indolent, perhaps the least aggressive form of the disease.
It is indeed. Smoldering ATL is the rarest and generally the most indolent subtype, affecting fewer than 5% of all ATL patients. In this type, while the malignant T cells still show evidence of monoclonal proviral integration, confirming they are cancerous, typically less than 5% of the peripheral blood lymphocytes exhibit those characteristic abnormal flower cell morphologies. Crucially, there's no hypercalcemia, no significant lymphadenopathy, and no hepatosplenomegaly, enlarged liver or spleen. The CNS, bones, and GI tract are typically not involved. However, mild skin lesions, like patches or plaques, and sometimes subtle pulmonary lesions may be present. The median survival for this small subset of patients appears to be 5 years or even longer, making it the most protracted and least immediately aggressive form. But again, like the chronic form, it still carries some risk of eventually progressing to acute ATL over time, so surveillance remains important.
That's an exceptionally detailed overview of the spectrum of ATL and its varied presentations, from explosive acute disease to the slow smoldering form. Now, let's turn our attention fully to the other major disease definitively associated with HTLV-1. Tropical spastic paraparesis, also known as HTLV-1-Associated Myelopathy, or HAM. How does its epidemiology, who gets it, and its typical clinical onset differ from what we just heard about ATL?
Right, HAM presents quite differently from ATL in several key aspects. First, in stark contrast to ATL, where there's a slight predominance of male patients, HAM disproportionately affects female patients. The reasons for this gender bias aren't entirely clear. Second, its clinical onset is typically insidious.
Meaning gradual.
Yes, very gradual and subtle, often developing over months or even years, unlike the rapid onset of acute ATL. Key initial symptoms that patients might notice often include a progressive sense of weakness or stiffness, usually beginning in one or both legs. Back pain is also a common early complaint, and often urinary incontinence or other bladder symptoms like urgency or frequency develop due to neurological involvement of the bladder control pathways in the spinal cord. Sensory changes like numbness or tingling are usually mild or less prominent than the motor deficits, but a superimposed peripheral neuropathy can also develop in some patients over time.
And how does the disease typically progress once it starts? And what are the usual long-term functional outcomes for patients living with HAM?
The disease generally takes the form of a slowly progressive and usually unremitting thoracic myelopathy. This means a progressive dysfunction of the spinal cord, primarily affecting the nerve tracts in the thoracic mid-back region that control the legs and bladder. It's a sobering reality that impacts quality of life significantly over time. Some studies suggest that perhaps 1/3 of patients become bedridden within about 10 years of their diagnosis, and maybe around 1/2 are unable to walk without assistance, like crutches or a walker, by that point.
That sounds truly debilitating. Does it affect thinking or other parts of the nervous system?
Importantly, cognitive function thinking, memory, personality is usually spared in HAM. Patients' minds generally remain clear, and significant cranial nerve abnormalities, affecting eyes, face, swallowing, etc., are also unusual. The damage is primarily localized to the spinal cord tracts. On physical examination, patients display those classic signs of upper motor neuron damage originating from the spinal cord lesion, spastic paraparesis, stiffness and weakness of the legs, or eventually paraplegia, paralysis, along with hyperreflexia, exaggerated tendon reflexes, often ankle clonus, rhythmic involuntary muscle contractions when the foot is stretched, and extensor plantar responses, the abnormal Babinski sign when the sole of the foot is stroked. These are all textbook indicators of central nervous system pathway involvement, specifically the corticospinal tracts.
That sounds like a very characteristic neurological picture. What do imaging studies like MRI scans or actual pathological examinations of the spinal cord tissue reveal in HAM patients that might explain these symptoms?
MRI scans of the brain often reveal some characteristic, though often subtle, lesions, particularly in the white matter and also in the paraventricular regions, areas around the fluid-filled ventricles. More prominently, MRI scans of the spinal cord itself often show atrophy, shrinkage, and may show signal abnormalities indicative of inflammation or damage within the cord. Pathological examination of spinal cord tissues, usually obtained post-mortem, shows the underlying damage. There's typically symmetric degeneration of the lateral columns of the spinal cord. These columns contain the crucial corticospinal tracts, which carry motor signals down from the brain. In some cases, the posterior columns, which carry sensory information up to the brain, are also affected, explaining some of the sensory symptoms.
And what's causing this degeneration? Is it inflammation?
Yes. Microscopically, the spinal meninges, the membrane surrounding the cord, and the cord parenchyma, the nerve tissue itself, contain a chronic inflammatory infiltrate. This infiltrate is composed primarily of immune cells, particularly CD8 plus cytotoxic T lymphocytes, along with some CD4 plus T cells and macrophages. This inflammation is associated with demyelination, the destruction of the protective myelin sheath that insulates nerve fibers, and also direct axonal damage, which ultimately leads to the progressive neurological deficits seen clinically. It strongly suggests an immune-mediated attack on the spinal cord tissue.
So an inflammatory process, likely driven by the immune system attacking the spinal cord. Is the HTLV-1 virus itself found directly infecting the cells within the CNS, like neurons or glial cells? And how does the overall viral presence and the host immune response compare in AM patients versus those with ATL?
That's another interesting point of contrast. HTLV-1 is not usually found infecting the actual resident cells of the central nervous system itself, like neurons or oligodendrocytes, the myelin-producing cells. However, the virus, or at least viral proteins or RNA, may be detected within that small population of infiltrating lymphocytes, the T cells that have migrated into the cerebrospinal fluid, CSF, and the spinal cord tissue of ham patients.
So the virus is in the infiltrating immune cells, not the nerve cells themselves.
Primarily, yes, that seems to be the case. Now, a general rule of thumb when comparing the two main HTLV-1 diseases is that, paradoxically perhaps, HTLV-1 replication appears to be somewhat greater or more active in ham patients than in most ATL patients, especially late-stage ATL where TAX is often silenced. This means there might be more active viral gene expression and perhaps low-level production of viral particles occurring in the infected lymphocytes of ham patients. Correspondingly, ham patients tend to mount a stronger, more robust systemic immune response to the virus compared to ATL patients.
A stronger immune response in the disease that looks like immune attack.
Exactly. Antibodies to HTLV-1 are readily detected in the serum, but critically, they also appear to be produced locally within the central nervous system compartment in ham patients. Antibody titers, levels, in the CSF, are often found to be higher than those in the serum, which points strongly towards intraethical meaning within the CNS antibody synthesis by infiltrating B cells or plasma cells. In fact, in rare instances, a patient with ham might even be seronegative in their blood, but have detectable antibodies to HTLV-1 exclusively in their CSF, further emphasizing the localized immune activity.
That suggests a very strong compartmentalized immune response happening right there in the CNS. What are the leading theories then behind ham's pathogenesis, considering this intense immune activity seems to be driving the damage? Is it autoimmunity?
The destruction of the pyramidal tracts, which causes the motor symptoms, clearly seems to involve these infiltrating HTLV-1 infected CD4 plus T cells and the responding CD8 plus cytotoxic T cells. But the precise trigger and mechanism of the damage is still debated. It's worth noting again that a similar progressive myelopathy can also result from chronic HIV infection, or much less commonly HTLV-2 infection, suggesting there might be some shared neuropathogenic mechanisms among these human retroviruses, perhaps related to chronic immune activation or inflammation in the CNS.
So what are the specific theories for ham?
One prominent theory suggests that susceptibility to developing ham may be genetically related to the presence of specific human leukocyte antigen, HLA, alleles in the host. These HLA molecules are responsible for presenting viral peptides, antigens, to T cells. The idea is that certain HLA types might present HTLV-1 antigens in a way that leads to an overly aggressive or misdirected immune response, perhaps an autoimmune response targeting host nervous system proteins that resemble viral proteins, molecular mimicry, or maybe excessive bystander activation of immune cells within the CNS. However, while intriguing, insufficient definitive data currently confirms a strong, specific HLA association driving ham risk.
Any other leads?
Intriguingly, another line of evidence comes from antibody studies. Antibodies found in the sera of ham patients have been shown in vitro, in lab experiments, to bind to a specific neuron-associated host protein called heteronuclear ribonuclear protein A1, HNRNP-A1. Furthermore, these antibodies were shown to actually interfere with neurotransmission in these lab models. This finding lends support to a potential autoimmune component, where the immune system, perhaps initially triggered by the virus, mistakenly produces antibodies that attack the host's own nervous system tissue, contributing to the damage.
So it's still quite unclear then what precise factors ultimately influence whether an individual HTLV-1 infection will eventually cause any clinical disease at all, and if it does, what determines whether it will manifest as a neoplasm like ATL or an autoimmune inflammatory neurological disorder like ham. Are there any specific putative factors that researchers are actively investigating to try and explain this critical divergence in disease outcome?
You've hit on a core and still largely unresolved question in the field of HTLV-1 research. It remains frustratingly unclear what factors truly dictate whether a given HTLV-1 infection will cause any disease whatsoever in a particular individual, since most remain asymptomatic carriers. And if it does, what steers the pathogenic process towards cancer, ATL, versus neurological disease, HAM? Researchers are actively investigating several plausible factors.
These include potential subtle differences in the genetic makeup of different viral strains or subtypes, the inherent susceptibility conferred by particular host, major histocompatibility complex, MHE haplotypes, related to those HLA alleles we discussed, which influence how antigens are presented to the immune system. The specific route of HTLV-1 infection, remember, perinatal infection seems more linked to ATL risk, while transfusion or sexual transmission might be more associated with HAM risk. The overall long-term viral load or proviral load maintained in the infected individual. HAM patients often have higher loads than asymptomatic carriers. And also the qualitative nature, the specific type and balance of the HTLV-1 related immune response mounted by the host. For example, the balance between cytotoxic T-cell response and regulatory T-cell response. However, despite these compelling hypotheses and ongoing research, few definitive data are currently available to conclusively link any single factor or combination of factors to these specific divergent disease outcomes. It's almost certainly a complex interplay of multiple elements involving the virus, the host genetics, and possibly environmental cofactors.
It's a fascinating puzzle with huge clinical implications.
Okay, beyond the two major players, ATL and HAM, are there other putative HTLV-1 related diseases? Perhaps conditions less definitively linked, but still observed with higher frequency in endemic areas that might be related to the virus.
Yes, there are several other conditions where an association with HTLV-1 infection has been observed or suggested, particularly in highly endemic areas, although establishing a definitive causal link for these is often more challenging than for ATL and HAM, and the evidence remains less robust.
What sort of conditions? For instance, bladder dysfunction, often manifesting as urinary incontinence or urgency, is reported quite commonly in HTLV-1 infected women, even in those who don't meet the full diagnostic criteria for HAM. It suggests a spectrum of neurological involvement might exist. Also, in endemic regions, various chronic inflammatory and potentially autoimmune diseases have been reported with higher frequency in HTLV-1 infected individuals and have sometimes been attributed to the virus. These include conditions like chronic uveitis, inflammation inside the eye, infective dermatitis, a severe chronic eczema-like skin condition often seen in children, chronic pneumonitis, inflammation of the lung tissue, certain forms of inflammatory arthritis resembling rheumatoid arthritis, and polymyositis, an inflammatory muscle disease causing weakness.
But the link isn't proven for these.
Right. It is very important to emphasize that while these associations are noted epidemiologically, a definitive causal relationship between HTLV-1 infection and these specific illnesses has not been definitively established in most cases. More rigorous research is really needed to differentiate true causation from mere correlation or coincidence, or perhaps the virus acting as a trigger in genetically predisposed individuals.
So while associations exist and are being studied, the scientific community is still working diligently to solidify those potential causal links beyond ATL and A. Okay, shifting gears to prevention. What are the crucial public health strategies for preventing HTLV-1 infection, given its known transmission routes, mother-to-child sexual blood?
Prevention strategies are absolutely crucial, especially in those highly endemic regions, and they largely mirror the successful strategies used for other sexually transmitted and blood-borne viruses, particularly HIV. First and foremost, to prevent mother-to-child transmission, which primarily occurs through breastfeeding, women in endemic areas who are known to be HTLV-1 infected are strongly advised not to breastfeed their children, provided that safe and affordable alternatives like formula feeding are readily available and culturally acceptable.
Okay, avoid breastfeeding if infected. What else?
Second, to prevent transfusion-associated transmission, screening of blood donors for serum antibodies to HTLV-1 is now standard practice in many countries with significant prevalence, effectively eliminating this route of transmission where implemented. Third, reducing sexual transmission relies on the consistent practice of safe sex, including the correct use of condoms, particularly important given the higher efficiency of male-to-female transmission. And finally, preventing blood-borne transmission among injection drug users requires harm reduction strategies, crucially including the avoidance of sharing needles and syringes. These integrated public health strategies are essential for controlling the spread of HTLV-1 infection.
And for those unfortunate individuals who do go in to develop one of the HTLV-1 related diseases like ATL or HAM, what are the current treatment options available? And perhaps more importantly, are current therapies generally curative, or are they more focused on managing the disease and its symptoms?
This is a critical point to understand up front. It's important to stress that for the relatively small number of patients who do develop clinically significant HTLV-1 related disease, either ATL or AM, current therapies are generally not curative. Eradicating the integrated provirus from infected cells remains an unsolved challenge. Therefore, the primary goal of treatment is typically to manage the symptoms, try to induce remission in the case of ATL, slow the progression of the disease, extend life expectancy where possible, and maintain or improve the patient's quality of life. But achieving a definitive cure for either ATL or HAM remains an elusive goal for now, unfortunately.
Okay, let's start with the ATL treatment then. How are the aggressive, acute, and lymphomatous types typically managed given their rapid progression and the challenges like drug resistance and opportunistic infections?
Right. The acute and lymphomatous types of ATL are particularly challenging to treat, precisely because they progress very rapidly and often occur in patients who are already somewhat immunocompromised. The immediate management often focuses on stabilizing the patient, particularly addressing life-threatening complications like severe hypercalcemia. Hypercalcemia is generally managed initially with aggressive hydration, medications like bisphosphonates or calcitonin to lower calcium levels, and often glucocorticoid administration like prednisone, which can also have some anti-tumor effect. Then, cytotoxic chemotherapy targeting the underlying T-cell neoplasm is the mainstay.
Is the chemotherapy effective?
While the ATL tumor cells are often initially highly responsive to combination chemotherapy regimens, similar to those used for other aggressive non-Hodgkin lymphomas, there are significant challenges that limit long-term success. As we discussed, these patients are highly susceptible to overwhelming bacterial and opportunistic infections due to their underlying HTLV-1-induced immunodeficiency, and infections are a major cause of death during or after chemotherapy. Furthermore, even if remission is achieved, ATL relapses are unfortunately very common, typically occurring within a relatively short period, often just 4 to 10 months after completing initial therapy in most cases. This makes achieving durable long-term remissions very difficult.
That's a very challenging prognosis indeed. What about specific drug regimens that have been tried? You mentioned earlier the combination of interferon alpha and zidovudine, AZT. Has that shown durable benefit?
The combination of interferon alpha, an immune modulator, and zidovudine, AZT, an antiretroviral drug, has shown some promise, particularly perhaps for the less aggressive chronic and smoldering forms of ATL, and maybe as maintenance therapy after initial chemo for acute lymphomatous types. Here's that critical insight again regarding zidovudine's mechanism in this context. It is likely effective primarily through its cytotoxic effects on the dividing malignant T cells. As a chain-terminating thymidine analog, it interferes with DNA synthesis and proliferation. Its efficacy here is thought to be more related to these direct anti-cancer properties rather than its antiviral effects against HTLV-1 itself, as active viral replication isn't clearly the main driver of ATL progression, unlike an HIV infection where AZT targets reverse transcriptase.
Has this combo led to cures?
Selected studies, particularly pioneering work from groups in France and Japan, have reported encouragingly high response rates and even suggested a potential for long-term survival. For example, a 40% 5-year survival rate in some analyses with this combination, especially in certain ATL subtypes. However, this level of sustained response and long-term benefit has not been universally replicated in all subsequent studies or clinical settings, and remains an area of active research and debate regarding its optimal use.
Are there other, perhaps more intensive chemotherapy programs or treatments like bone marrow transplantation being used for ATL?
Yes, more intensive approaches have certainly been explored, particularly in Japan, where ATL is more common. LSG-15 is a specific complex multi-drug chemotherapy program developed in Japan specifically for ATL. Studies have shown it can induce complete responses in about 1/3 of patients with aggressive ATL, with approximately half of those responders surviving for over 2 years. However, even with this intensive regimen, the median overall survival time for all patients treated is still relatively short, only about 13 months on average.
What about transplant?
High-dose chemotherapy, followed by allogeneic hematopoietic stem cell transplantation, getting stem cells from a healthy donor, has also been widely tested, again mainly in Japan. While large studies haven't shown a significant improvement in the median survival for the overall patient population compared to chemotherapy alone, it has shown some promise in achieving long-term durable remissions in a subset of patients. Perhaps up to 25% of selected patients undergoing transplant achieve a disease-free survival of 4 years or more, suggesting it can offer a potential curative option for a minority of younger, fitter patients with a suitable donor, although it carries significant risks.
What about newer targeted therapies for ATL? Are there any promising agents emerging? And how are the less aggressive chronic and smoldering forms typically managed?
Newer targeted therapies are definitely emerging and showing some promise, offering hope beyond traditional chemotherapy. Lenalidomide, an immunomodulatory drug known for its effectiveness in multiple myeloma, has been reported to achieve a respectable 42% overall response rate in patients with relapsed or refractory ATL, extending the median overall survival in that difficult setting to around 20 months, although the duration of the response, progression-free survival, was shorter, around 4 months. Mogamulizumab is another important agent. It's a monoclonal antibody that targets CCR4, a chemokine receptor that is frequently highly expressed on the surface of ATL cells.
Yeah.
Clinical trials have shown that adding mogamulizumab to standard chemotherapy regimens can improve response rates and potentially survival outcomes.
Any other experimental approaches?
There are also promising experimental avenues being explored, such as developing antibodies conjugated to radioisotopes, like yttrium-90, or potent toxins, designed to target proteins highly expressed on ATL cells, like the IL-2 receptor, CD25. The goal is to deliver a lethal payload directly to the cancer cells while minimizing damage to normal tissues, though these approaches are not yet widely available clinically.
And for chronic smoldering ATL?
For the much less aggressive, chronic, or smoldering forms of ATL, which might remain stable for years, the management strategy is often quite different. It frequently involves an expectant approach, sometimes called watchful waiting. This means actively monitoring the patient for any signs of disease progression, like rising lymphocyte counts, worsening skin lesions, or development of hypercalcemia, and promptly treating any intercurrent opportunistic infections as they arise, but often withholding aggressive chemotherapy until there's clear evidence the disease is progressing towards a more acute phase. Sometimes the interferon-AZT combination might be used in these settings too.
So a range of treatments tailored to the subtype and aggressiveness, but still a very difficult disease to cure overall.
Okay, what about treatment for HAM, the chronic neurological condition? What options exist there?
For HAM, the treatment landscape is quite different, as the goal isn't killing cancer cells, but rather modulating the chronic inflammation in the spinal cord and managing the resulting neurological symptoms. Patients with HAM may derive some symptomatic benefit, particularly in reducing spasticity or perhaps slowing progression early on, from treatment with glucocorticoids, such as oral corticosteroids like prednisone or intravenous methylprednisolone, which help reduce inflammation. However, long-term steroid use has significant side effects, limiting their utility.
Do the antiretrovirals that work for HIV help? Importantly, and perhaps surprisingly, conventional antiretroviral regimens that are highly effective against HIV, like reverse transcriptase inhibitors or protease inhibitors, have generally not been found to be effective for improving neurological outcomes in HAM. This again suggests that ongoing viral replication might not be the primary driver of the neurological damage, which seems more related to the chronic inflammatory response.
Are there any other drugs that have shown promise?
There have been some interesting findings in smaller studies, though they need confirmation. One older study, for example, recorded significant neurologic improvement in 5 out of 6 HAM patients who were treated with danazol, which is an attenuated androgen, a synthetic steroid hormone, at a dose of 200 milligrams 3 times daily. Remarkably, 2 patients in that small study even reversed their confinement to a wheelchair. More recently, an antibody targeting the IL-15 receptor beta chain has also been tested in small numbers of patients with some promising clinical effects reported, hinting that the cytokine IL-15 might play a role in driving the damaging immune response in HAM.
So, limited drug options, mostly focused on inflammation. What else is important for managing HAM?
Given the limited efficacy of drugs in reversing established damage, supportive care becomes absolutely paramount in managing HAM. Physical therapy and rehabilitation programs are absolutely vital components of care. These aim to help patients maintain muscle strength and function as much as possible, manage spasticity, improve mobility, using assistive devices if needed, prevent complications like contractures or pressure sores, and significantly enhance their overall quality of life as they cope with the progressive neurological deficits. Managing bladder dysfunction with medications or other strategies is also critical.
That's a very comprehensive look at HTLV-1, its associated diseases, and the current state of management. Now let's briefly touch upon the other known human T-cell lymphotropic virus, HTLV-2. You previously referred to it somewhat cryptically as a virus searching for a disease. What's its story, and how does it compare epidemiologically and clinically to HTLV-1?
Right, HTLV-2. It was isolated just 2 years after HTLV-1 back in 1982. Also from a patient with a hematologic malignancy specifically, an unusual T-cell variant of hairy cell leukemia. This initial discovery again suggested a potential link to blood cancers. Epidemiologically, HTLV-2 has a different pattern than HTLV-1. It's found to be endemic primarily in certain indigenous Native American populations, particularly in North, Central, and South America. It's also found sporadically in parts of Africa, particularly among pygmy populations.
And its origins.
It's generally considered a New World virus, distinct from the Old World origins of HTLV-1. The prevailing hypothesis is that HTLV-2 may have been brought from Asia to the Americas tens of thousands of years ago, perhaps between 10,000 and 40,000 years ago, during the migration of infected human populations across the ancient Bering Land Bridge that connected Asia and North America.
Fascinating ancient history, possibly preserved in a virus. How is HTLV-2 transmitted and what's its prevalence like, particularly among specific risk groups like injection drug users?
Its mode of transmission is largely believed to be the same as HTLV-1. Primarily mother to child, especially via breastfeeding seems important, sexual contact, and bloodborne routes, sharing needles, transfusion. However, some epidemiological evidence suggests it might be somewhat less readily transmitted sexually compared to HTLV-1. Now, a key finding came from studies of large cohorts of injection drug users, IDUs, particularly in the US and Europe, especially once diagnostic assays became available that could reliably distinguish HTLV-1 from HTLV-2 antibodies.
What did those studies show?
They revealed, somewhat surprisingly at the time, that the vast majority of HTLV-positive individuals within these IDU cohorts were actually infected with HTLV-2, not HTLV-1. For instance, in a large U.S. cohort study involving over 7,800 IDUs across various cities, the overall HTLV seroprevalence was quite high, around 20.9%. But critically, more than 97% of those positive cases were found to be due to HTLV-2 infection.
Wow, so almost all the HTLE in that IDU population was HTLV-2. Any geographic patterns?
Yes. Within the U.S., seroprevalence was notably higher in the Southwest and Midwest compared to the Northeast, and co-infection was also seen. Approximately 3% of the members of that large cohort were found to be infected with both HTLV-2 and HIV-1, highlighting the significant epidemiological overlap in transmission routes within this population. Demographically, similar to HTLV-1, seroprevalence increased linearly with age. And interestingly, women were significantly more likely than men to be infected with HTLV-2, again suggesting potentially more efficient male-to-female sexual transmission.
So it's clearly quite prevalent in certain populations, particularly injection drug users in some regions. But you still describe it, or it's often described, as a virus searching for a definitive disease connection. What exactly does that mean clinically? Does it cause any specific disease?
That's precisely the central enigma of HTLV-2. Although it was initially isolated from that patient with a T-cell leukemia variant, and despite its relatively high prevalence in certain specific populations like IDUs and some Native American groups, HTLV-2 has not been consistently or definitively associated with a particular well-defined disease in the same clear and unambiguous way that HTLV-1 is linked to ATL and HAM.
So no HTLV-2 leukemia or HTLV-2 myelopathy widely recognized.
Not as distinct, clearly defined entities with strong, consistent epidemiological links like ATL and HAM have with HTLV-1. For this reason, HTLV-2 is often informally, though perhaps a bit dismissively, referred to as a virus searching for a disease. However, that doesn't mean it's necessarily harmless. While a clear signature disease remains elusive, evidence is accumulating, particularly more recently, suggesting that HTLV-2 infection may indeed play a role in, or at least increase the risk for, certain neurologic conditions, perhaps milder myelopathies or peripheral neuropathies, some hematologic abnormalities, and possibly certain dermatologic diseases, or increased susceptibility to infections like pneumonia or tuberculosis.
So potential links are emerging, but need more proof.
Exactly. It's important to emphasize that these potential links require further confirmation through large, well-designed cohort studies, especially given the past confusion in some older studies that couldn't reliably distinguish HTLV-1 and HTLV-2 infections, which may have clouded the epidemiological picture among IDUs, for example. It remains a very active area of ongoing investigation to clarify the true clinical impact, if any, of chronic HTLV-2 infection.
And for prevention of HTLV-2, I imagine the principles are largely the same as for HTLV-1, given the shared transmission routes.
Exactly right. Similar public health principles and strategies apply equally to HTLV-2 prevention as they do for HTLV-1. This includes crucial harm reduction strategies like avoidance of needle sharing among IDUs, adherence to safe sex practices including condom use, robust screening of blood products for transfusion, using assays for HTLV-1, which typically also detect HTLV-2 antibodies due to cross-reactivity, and advising infected women to avoid breastfeeding in situations where alternative feeding methods are safe, feasible, and culturally acceptable.
Okay. And finally, just to place it within this broader context of human retroviruses, we need to give a brief but essential mention to the human immunodeficiency virus, or HIV.
Absolutely. HIV-1 and its less common relative HIV-2 are arguably the most globally significant and certainly the most infamous members of the lentivirus subfamily within the broader Retroviridae family. They are, as far as we know, the only lentiviruses that naturally and commonly infect humans, causing acquired immunodeficiency syndrome, or AIDS.
And the term lentivirus itself tells us something.
Yes, the very term lentivirus is derived from the Latin word lentus, which means slow. This name implies a slower-acting or chronic type of virus, contrasting sharply with viruses that cause acute, short-lived infections like influenza or the common cold. The characteristic chronicity and often very long latency period of untreated HIV disease, where it can progress slowly over many years, gradually destroying the immune system, is perfectly consistent with its designation as a lentivirus. It establishes a persistent, lifelong infection that the body cannot clear on its own.
And we're not diving deep into HIV today.
Right. While a detailed discussion of HIV biology, pathogenesis, treatment, and prevention is an enormous and critically important topic in itself, comprehensively covered in Chapter 202 of our source material, the next chapter we acknowledge its immensely important place within the retrovirus family, as undoubtedly the most significant human pathogen discovered in this class, having caused a global pandemic.
What an incredible and incredibly detailed journey we've taken today.
Yeah.
Seriously, from the precise molecular mechanisms of how influenza antivirals work, right through to the truly mind-bending, paradigm-shifting biology of retroviruses, we've really gone deep into how these microscopic entities challenge and continually redefine our fundamental understanding of life itself.
We really have. We've thoroughly explored the unique nature of retroviruses, particularly their revolutionary ability to transcribe RNA back into DNA using reverse transcriptase, and the profound implications this discovery had for our understanding of molecular biology, literally overturning that central dogma we all learned. And we've seen in detail how these complex viruses, particularly HTLV-1, can lead to such diverse and severe diseases in humans, ranging from those aggressive cancers like adult T-cell leukemia lymphoma to chronic autoimmune or inflammatory conditions like HTLV-1-Associated Myelopathy, each with their own intricate and still partially understood pathogenesis.
And it wasn't just about the diseases, was it? We also meticulously dissected their sophisticated life cycles, really understanding how they involve these intricate interactions with our own host cells, driven not just by their core structural genes, GAG, POL, and B, but crucially by those incredibly complex and strategically vital accessory genes like TAX, REX, HBZ, and HTLV, or TAT, REV, NEF, VIF, VPR, VPSC, and HIV.
Exactly. And gaining that granular understanding of these precise molecular dances, how the virus manipulates the host, how the host fights back, how the virus evolves to evade those defenses, is what truly elevates one's grasp of the entire field. It moves you beyond just memorizing facts to achieving a genuine professional level mastery of the concepts.
Indeed. Cultivating this kind of deep, almost mechanistic understanding helps us appreciate the extraordinary complexities of disease pathogenesis, why developing truly curative treatments for these chronic viral infections is so challenging, and even glimpsing the immense potential and the inherent risks of carefully harnessing these unique biological tools in cutting-edge fields like gene therapy.
It really is a field defined by continuous discovery, profound biological insights, and significant ethical considerations, especially as we learn to manipulate these powerful biological agents. It's a testament to the continuous evolution of our scientific knowledge and the incredible, almost unbelievable intricacies of life operating at the microscopic and molecular level.
Absolutely. And to leave you, our listeners, with a final provocative thought, something to perhaps mull over and to continue your own deep dive into this fascinating subject long after we finish talking.
Consider this. How does the fundamental ability of retroviruses to integrate themselves into our very own genome, a process once thought biologically impossible, continue to challenge our understanding of biological information flow and the very definition of self versus non-self at the genetic level? This unprecedented genetic intertwining opens doors, both incredibly promising for therapy, but also potentially precarious for future genetic interventions. What further secrets might these viral time travelers, these ancient genomic invaders, hold about our own human evolution, our susceptibility to diseases like cancer and autoimmunity? And how might our continued deep dive into their mysteries, unraveling their complex strategies, profoundly shape the future of medicine and biotechnology?
That's a lot to think about. Thank you for joining us on this very deep dive into the world of influenza, antivirals, and especially human retroviruses. We sincerely hope you feel not just informed, but genuinely enlightened, perhaps a little awed by the complexity, definitely curious, and maybe even empowered to explore these profound biological mysteries even further. Until next time on The Deep Dive.